Now, I would like to ask Dr. Takahashi, Chief Executive Officer, LINK-J, and Dean Albert P. Pisano from UC San Diego, University California, San Diego, to give opening remarks to the audience. Firstly, Takahashi-san, please.
Hello, everyone. Thank you very much for joining us today. Before we begin, I'd like to take a moment to express my sincere appreciation to everyone joining us from overseas. In particular, I'd like to thank Dean Pisano from UCSD, who is kindly serving a moderator today, and also Professor Alexandrov and Moazan and Nakagawa and the other distinguished speakers. And also, I'd like to express my special thanks to Miwako Waga-san from UCSD, for her invaluable support in organizing this event. Thank you very much. With that, I'd like to continue in Japanese for my official opening remark. 皆さん、こんにちは。LINK-J の高橋です。本日も多くの皆様にお集まりいただきありがとうございます。先ほどもありましたけれども、今回第8回 LINK-J UC サンディエゴライフサイエンスシンポジウムゲノム解析 AI 個別化医療次世代バイオマーカーとの医療応用ということで、第8回ということにもありますように、UCSD と我々 LINK-J との間での合意にも続きまして年に1回このように日本にもう著名な先生方にお越しをいただいてこういうような機会をご提供できるということも我々 LINK-J としても大変嬉しく思います。で、我々 LINK-J がちょうどこの春に10年の節目を迎えました。そういう意味におきましてはこの UCSD と我々の LINK-J との協業というのがもうその大部分にわたって続いているということも我々は本当に嬉しく思いますし、実はこのオープニングの直前にですね、ディーン・ピサノと我々 LINK-J、私の間で新たなMOU の継続というものにちょうど署名をしたところです。そういう意味でこれからも我々はUCSD の皆さんともにですね、日本の研究者にホットなサイエンスの話題を提供できるということをお約束をいたします。さて今日のイベントなんですけども、AI、それからゲノム解析、個別化医療、これいずれもですね相互にかなり親和性が高いトピックです。で皆様ご存知のように、製薬産業におきましても、いろんな研究で AI の活用というのが急激に進んでおりますし、大きなディールもかなり昨年来増えているというところです。そういう中で今日のトピックとはまさにホットな話題ということでですね、今日冒頭300名というお話もありましたけれども、多くの皆様に興味をいただいてご登録をいただいています。でオンラインの方々が多いっていうのは事実なんですけども、我々のイベント後にですね、もう登壇の皆様とのネットワーキングの場というものを用意しています。今からでも間に合いますので、ご近隣の方々はぜひ会場にお越しいただければいいんじゃないかなというふうにも思います。ということで、今日本当に楽しいお話いろいろ伺えると思いますので、楽しいというと語弊がありますかね、素晴らしい研究の成果、また今後につながるお話をいただけると思います。ぜひご来場の皆さんもその後のネットワーキングを含めて、活用していただければというふうに思います。本日はどうぞよろしくお願いします。
Thank you, Takashi-san. Next, Dean Pisano, please.
Konnichiwa, everybody. Thank you, Mr. Kiyuchi. Thank you, Dr. Takahashi. My name is Al Pisano. I'm the Dean of the Jacobs School of Engineering. And as we just heard, the Jacobs School and LINK-J have been partners for a long time, and we're happy to be continuing that. We have been happy we have been pleased to be part of LINK-J, and we are happy to be organizing these talks and panel sessions because it connects Japan and the United States together, as well as Japanese academicians and industry members with the University of California, San Diego. I want to give special thanks to Ms. Miwako Waga for all her efforts and her organizational skills. If it went well, it's her credit, and if it went badly, it's my fault. So I hope we all understand how to assign the blame. Good there, bad here. Okay, good. All right. Thank you to our speakers, and we're going to have 4 very dynamic speakers. We are all eager to hear your wonderful insights, and we all wish to learn from you, me included. I will be taking notes. I will conclude my opening comments by making 4 observations. Observation number 1, we will hear about many wonderful technologies this afternoon. 2, we will hear about much wonderful science this afternoon. 3, we will hear about the medical innovation ecosystem. But the most important point I want to make is number 4. The real challenge is for us to bring all of these things together. We must bring them together in such a way that they can be integrated into medical workflows, and so that we can achieve better patient outcomes at lower cost. So we need better outcomes and lower cost. So these objectives, improved outcomes, lower cost, are necessary for the future of medicine, and I'm very happy and eager to hear what our wonderful 4 speakers have to say. Thank you.
Thank you, Dean Pisano. Now let's move on to the main part of the seminar. We are honored to have a keynote presentation from Professor Radmil B. Alexandrov, Professor, Department of Cellular and Molecular Medicine, and Shu Chen Jin Lei, Department of Bioengineering, UC San Diego. Professor Alexandrov, please.
Thank you so much, and thank you so much for having me. I'm delighted to be back in Tokyo and to participate in this LINK-J special symposium. I hope that today's day is going to let us think more about genome analysis, more about AI, and more about cancer treatment and prevention. And my talk has 2 parts. In the first part, I'll tell you how to use AI for cancer treatment and cancer prevention. And in the second part, I'm going to tell you how to use AI for precision diagnostics. Before I start, these are my conflicts of interest that I'm required to disclose. Please keep them in mind when I'm presenting. So the first part of the talk, we're going to focus on genome analysis and AI for preventing cancer, and specifically for preventing early onset colorectal cancer. And before I start talking about colorectal cancer, I'll tell you about somatic mutations, mutational signatures, and how they connect to human cancer. And mutations accumulate daily in every single cell of our body. They accumulate because the cells behave properly, because the cells divide. They also accumulate because of the lifestyle choices we make, smoking tobacco, cigarettes, eating certain types of food. And they may accumulate because the cells do not function correctly, such as defects in DNA repair. Now, we have known for a long time that cancer risk is strongly related to somatic mutations. The more mutations, the higher the risk for cancer. And the example for that is lung cancer and tobacco smoking. And if you take one subtype of lung cancer, lung squamous cell carcinoma, from 105 patients, 1 is a never smoker. And usually that person has most likely smoked earlier in their life and they have misreported their smoking habits. So if smoking didn't exist, lung squamous cell carcinoma would be most likely a non-existent tumor type. About 10 years ago, 10 or 15 years ago, the question came whether can we say what caused the mutation in a person by sequencing the genome of the cancer? And essentially, this gave rise to something called mutational signatures, where we can sequence cancer genomes and being able to identify the mutagenic agents that cause this cancer. And let me give you an example. Skin cancer, we all associate with ultraviolet light exposure because most skin cancers are caused by UV light. The UV light exposure generates a specific mutational signature, specific pattern of mutations that you can see on your screen, these red peaks, C2T mutations at dipyrimidines. If you get exposed to UV light, these are the mutations your genome will get peppered with. And then you can think of lung cancer. And from all types of lung cancer, 80% of them are caused by tobacco smoking. And this is the mutational signature of tobacco smoking. It generates many, many mutations, which are C2A or T2A. And again, it generates many of the mutations that drive lung cancer. Over the last 10 or 15 years, we and others have found many mutational signatures. Now there are over 100 of them. Some signatures are associated with processes within the cells. Others with treatment. The fact that you treat cancer can cause cancer by itself. And lastly, there are environmental exposures. There are many environmental exposures that can cause cancer, and I give some examples here on the screen. Now, we have over 100 different mutational signatures, and for about half of them, we understand what causes them. One signature very relevant for this talk and very relevant for colorectal cancer is the signature of colibactin. And colibactin is something that you may not have heard of. It happens to be a secondary metabolite generated by a bacteria that has specific locus in the genome called the PKS island. And the bacteria has that to fight other bacteria. It generates genotoxins that attack the genetic materials of other bacteria surrounded. But if you have colonization, if you have that bacteria in your colon, it will mutate your colon, and it may increase your risk for cancer. In 2020, Hans Clevers and colleagues in the Netherlands performed this experiment. They exposed organoids to the bacteria with the PKS island and without. And they found a mutational signature that they called the PKS mutational signature. Very clear, very precise. At the same time, we and others were sequencing normal colon tissue and normal colorectal cancers. And we found mutational signatures that we gave a number, 88, because we didn't know what's caused. Now, based on similarities and everything we know, this is the signature of colibactin. This is the signatures of you having that bacteria. And as it turns out, and as I'll show you, it becomes a very important signature for what we think causes colorectal cancer. Now, because mutational signatures and these patterns of mutations can tell you the causes in colorectal cancer, that can allow you to do something called genomic epidemiology. You can go around the world and try to understand why people get cancer or why different countries have different risks for developing cancer. And we were very lucky in 2017 to get $25 million Cancer Grant Challenges process to do that, to go to 40 countries around the world and to try to understand why some countries get more cancers than others. One of these cancers was colon cancer. And colon cancer is a very, very interesting case. You can see the incidence rate of colon cancer here from the United States. If you're over 50, the incidence rates, the amount of colon cancer you get in the population are going down. If you're under 50, the incidence rates are going up. And the younger you are, the more colorectal cancer you can have. This is true for the US. This is true for other countries. Now, the risk for colon cancer is also different around the world. In fact, Japan has one of the highest risks for colon cancer in the world. But also, has the increase in this early onset cases. We have seen twofold increase in the US for the early onset cases. And again, this was first reported in 2019, subsequently in Australia, Canada, Japanese, a number of European countries, and now we know this is happening in 27 countries around the world. There are no currently any known risk factors. We do not know what causes this increase. But it's not only an increase, it is now the leading cause of cancer death in young adults. It is the thing that kills most people under the age of 45. And the younger the people, the higher, the more of them die, the higher percentage of them die. So it's a very big health issue, a global health issue, if you wish. And there have been many reports on that topic, telling you that's important, telling you, giving examples of people getting colorectal cancer, usually young, usually very, very fit. Now, what the study that we did is to collect colon cancers from 1,000 patients, and for each patient we got the cancer and blood. We did it in 11 countries around the world with different risks for developing cancer. And you can see here Japan with the highest risk for cancer, and you can see countries like Iran and Thailand with much lower risk for developing cancer. The purpose of our study was to understand what causes cancer, to be able to compare what causes cancer in one country versus another. But just because early onset cases were increasing so much, we had 132 cases within our cohort. And you can see that they're very nicely represented across all the different subtypes of colon cancer, whether it's the proximal colon, the distal colon, or the rectum. There are many ways to show colon cancer, and because I'm a cancer genomicist, I'm going to show it to you in the most genomics possible way. What you see here is on this plot is the number of single point mutations. These are single bases of DNA that are being changed, and that's on the y-axis of these plots. And on the x-axis, you see per part of the genome of the tumor that has been affected, whether the cancer is stable or unstable, and you also see the small insertions and deletions. And the reason I'm using these molecular biomarkers is to show you that colon cancer is actually different molecular diseases. And the first one is something what we call microsatellite stable or DNA repair proficient colon cancer. It can have very stable or very unstable genome, and it will have about 10,000 mutations on average. Now, this cancer is about 80% of all the colon cancer. But it's very, very different than the next big subtype, the microsatellite unstable tumor. There, you have a failure of a repair process. You have skyrocketing mutations, an order of magnitude for single point mutations, and 2 orders of magnitude for these insertions and deletions. This is a tumor that will give immune checkpoint inhibitors when we treat, and usually the patients are going to respond quite well. It's about 15% of all colon cancer cases. And there are many other subtypes related to polymerase deficiencies, failure of different types of DNA repair, but they are usually less than 1% of the colon cancer. They look different, they have different treatment regimens, but they're more of the exception rather than the rule. And in these cancers, again, we have some failure of some repair mechanisms that has usually led to the cancer. Now, when we were collecting our cohort, it was supposed to be treatment naive before patients received treatment. But in 2 patients, we found the evidence of treatment, the mutational signatures of treatment. And it turned out that what has happened is these people had cancer when they were children. That cancer was treated heavily with chemotherapy, and the signatures, the mutations from childhood tumors, were still present in their adulthood tumors. Now, for all purposes and analysis, all these categories that have a handful of samples were excluded. We reported them and moved because you cannot do much with just that few samples per category. We finalized the microsatellite unstable tumors and didn't find anything. And the main work we have done is these tumors that are stable, that do not have a failure of DNA repair. These are the tumors where you can see what's the environmental exposure that causes them. We did our usual analysis of mutational signatures, and we found the usual suspects. We found the signatures that accumulate with age. We found the signatures due to endogenous deaminases, the ones due to reactive oxygen species. We found the colibactin signature, and that's going to become important in just a second. And then we found many, many signatures that we do not know what causes them. We know they're important. We know that may have explained the reason we get cancer around the world, but we do not know their etiology. And again, this is just one type of mutational signatures. There are many, many other types of signatures that we also analyzed. So the first question one may ask is, did you observe any difference between countries? We have different cancer risk. Are there differences of mutagenic exposures? And we did the proper comparisons. We did a comparison where one country was compared to all the others, and we looked whether there are any differences. And we did find differences. In Russia, we found two signatures that are highly enriched, one of them unknown and one of them due to endogenous processes. In Argentina, we found three signatures that are enriched, all of them of unknown etiology. And in Brazil, one that's enriched. In Colombia, three that are enriched and one that's depleted. And in Thailand, one that's depleted. But again, there is not much we can say about these signatures. We know they're there. We know that they're the fingerprints left from exposures, but we don't know what causes them. And we can see how, you can see here how they look on a map. You can see one of those signatures very highly enriched in Colombia, also present at lower levels in other parts of the world. And the other one very much enriched in Argentina and also very much at lower levels around the world. And the question comes, what about the mutational signatures in early onset cases in this young individual? And our study was the first to show that there are big differences in these young people versus the old. And what we showed very clearly is that there is at least three mutational signatures enriched in the early onset cases. And these were the signatures of colibactin and one signature of unknown etiology. So the signatures SBS88, ID18 were very, very highly enriched in the early onset cases. To give you a comparison, one of every two patients under the age of 40 will have these signatures. And if you look at older patients, over 70, it will be less than 15%. So you're talking about 3 to 4 times enrichment. The question came, do they have the bacterial infection? Do they have actually the bacteria there causing them? And this is one patient, and we can look at them, and they very, very clearly have that bacterial island. And then this is another patient. They have, again, these mutational signatures, but they do not have the bacterial infection. And when you do it across the whole population, you will see that some of the patients have it and some don't, but there is no associations of seeing the bacteria and seeing the mutations from that bacteria. And that's true by age group. And the reason for that is you can have the bacteria there generating the mutations in the past. Remember the chemotherapy treatment. You have people treated as children, the chemotherapy stays there. So if these people have had at one point of their life infection of these bacteria, then this would have stayed. So then we started hunting down, when did this happen? And again, we did a number of fancy mathematical modeling to understand when did it happen in the evolution of the tumor, and it happened very, very early in the evolution of the tumor. That doesn't tell you at what age, but it tells you it's one of the first events. The next thing that we did is to try to understand when did it happen in a person's life, right? When is in the lifetime of the person when this has happened? And to do that, you need to do a bit of molecular archaeology. For each cancer patient, we needed to get normal colorectal tissue, colorectal crypt, sequence each one of them, and put them on phylogenetic trees. You can think this as the way we time species, the way we have timing how apes diverge from humans, for example. We have put some molecular timers, we have put them on a phylogenetic tree, and we can see when these things have separated. And again, we did exactly the same. For this one patient, we sequenced 15 colorectal crypts, we put them on the phylogenetic tree, and we assigned the mutational signatures. And what we saw here, what you see in this example here, is two colorectal crypts, and they have diverged, but they have a shared branch. And that means they had a common ancestor, something that came before them. And you can see that the pink color, that colibactin, is in the common ancestor. That means the exposure is early, And that means the exposure happened before they diverged. And as you can see, none of the leaves actually have the pink color. So you have exposure that happens early, and then it stops. And now you can use the different fancy molecular timers to say, at what age do we get the signatures? And our timing indicated the first 5 years of life. So we're talking about an infection in children in the age before they're 5. Now, the question whether this is causing mutations, but we also ask the question, does it cause the mutations that drive cancer? And the striking result we got there is APC. The gene APC is one of the first hits in colorectal cancer development. And a substantial proportion of all the APC mutations were caused by the infection or by the presence of these PKS positive bacteria. So you don't only have them generating mutations, you have them actually generating the mutations that drive cancer. So I'm going to summarize this part of the talk, and then I'm going to tell you our hypothesis of what causes early onset cancer. We have gone to 11 countries, and we have gotten samples from 1,000 patients, including 132 early onset cases. We see all the known molecular phenotypes for colorectal cancer. We have found over 47 mutational signatures in these countries that will likely explain the difference in cancer risk. Most of them, we don't know their etiologies, and I'm also delighted to say that as of May of this year, we have gotten another grant challenge for $25 million to hunt the origins of every single one of those signatures. We see multiple mutational signatures in the early onset colorectal cancer cases. We see the colibactin there, but we see others that we don't know what they are just because we don't know their etiology. There is no evidence that you have the infection at the moment of cancer diagnosis. And again, when you time these mutational signatures, they're early in the tumor, and they most likely happen in the first 5 years of life. And they generate some of the first mutations that drive tumors. So you can summarize these things in a hypothesis. You can say, well, what causes colorectal cancer? Or our hypothesis of what causes colorectal cancer is infection in very, very early life with this bacteria that goes and mutates the colon tissue of children, and generates some of the first driver mutations in these children. And essentially, if you get the driver mutation in a child of age 5 or 4, well, now they're 20 to 30 years ahead of schedule to get that tumor. We see it for colibactin. We think there's at least 2 other signatures that behave in exactly the same way. Now, every hypothesis needs to have a bit of additional support. And one of the things we always felt queasy about is whether this really happens in children. We needed to time it, we needed to use models. And essentially what we decided to do is, can we get children from actual tissues from actual children? And this is exactly what we did. These are children from UK and Denmark. These are children that have gone to a gastroenterologist and have gotten a colon biopsy, so they're not unbiased. Something has happened in these children. But they have gotten a clean bill of health. They have, based on the medical advice, they do not have anything in their colon. In 40% of these children, we see the mutations in colibactin. All of these children are under the age of 18. So by the definition, children. Many of them are under the age of 5. So we can actually see that in most cases, it happens in the age of 5. So our hypothesis is that you have cases that you have these bacteria that generates these mutations, and it generates very few mutations, and it probably doesn't increase the risk for colorectal cancer, like this child here. But there are others where you have many, many mutations. And this child is about, I think, just about 2 years old. And the colon of this child looks at about somebody in their late 50s. So we think that this child will be at risk for developing colon cancer very early in their life. Unfortunately, we cannot follow these children, and we don't know whether they have this information due to consent issues. And again, this is children in Europe. We think in the US, the proportions will be similar. We actually think in Japan, they will be even higher. But again, we think the exposure, we're talking about population level exposure at about 40% of the population. And again, for some outliers leading to early onset colon cancer. So we have summarized that in a very short piece for cancer discovery with my partners in intellectual crime, Mike Stratton and Paul Brenner. It's about 1,200 words piece where we say, well, this is what we think, this is what the evidence is, and this is how we prove or disprove this evidence. So if you're interested about that, there is a QR code here. With that, I want to thank the people who did the work for this part of the talk. Marcos Diaz-Gay, who was a postdoc at UCSD and now has his own lab in Spain. And again, our close colleagues at the Sanger Institute, who did a lot of the sequencing, and Sarah Moody was the co-first author, and our colleagues in the International Agency for Research on Cancer, who did the actual physical collecting of this over close to 1,000 samples. And also, he's not here, but Tatsuhiro Shibata, who was actually the person who helped us get samples from Japan and being able to see what happens in the Japanese population. And again, this was funded by the Cancer Grant Challenge, the Mutagraphs Cancer Grant Challenges. So this is my first part of the talk. This is in the first part, I showed you how you use machine learning and AI to figure out what causes cancer and puts you on the pathway to try to potentially prevent cancer. The second part is to talk about precision oncology. Now you have cancer. How can you better treat that cancer, or how can you better deal with it? And this was done by Eric Bergstrom, who was a postdoc, PhD first, and then postdoc in my lab, and now has moved on into industry. So usually, when you look at cancers, it gets next-generation sequencing profiling or molecular sequencing. However, next-generation sequencing or molecular profiling, this is not accessible in the US to all patients, and the limit outside of the US and outside of highly developed economic areas is very, very limited. And just to give you an example, in lung cancer, there was the FDA-approved biomarkers in 2004. It's 22 years later, less than 50% of the population in the US takes that molecular test. We know that they will get better treatment. We'll know that they will get better survival, but more than half of them do not get it. And there's many reasons for that, from how the US health system is structured to the actual details of how these biomarkers are actually detected. And in the US specifically, we see very big disparities between different ethnic groups, different socioeconomic status, and essentially different groups of people. Now, let's look at the somewhat simplified but a real workflow of how cancer gets treated. Imagine you detect a lump. Let's talk about breast cancer. You detect a lump of your breast. It takes a few weeks to see your primary care physician to say, hey, I have a lump. That person will refer you to oncology, which may take another 3 to 4 weeks. In oncology, they'll take a biopsy, they'll do pathology diagnosis and staining, and in about 3 to 6 weeks, you will know, do you have tumor, do you have benign tumor, or do you have malignant tumor, and what kind of tumor you have if you have one. So now you're in the pipeline, but you're about, 2 months after you have found that lump. You go to your oncologist, and the oncologist tells you, we need to do molecular profiling to be able to give you the best treatment. You as a patient always would say, okay, I want the best possible treatment. Well, that means the pathologist writes a report. So the oncologist tells the pathologist to send, to actually get some of the tissue and to send it to molecular profiling. That by itself can take 2 months. Now, in about 30% of cases, the molecular profiling will fail depending on the test. So you either need to do not precision oncology, just do essentially random guessing, or you need to repeat that procedure, another biopsy, another molecular profiling, and in the end, you get a report. Now, in some cancer cases, this works well. In pancreatic cancer, we don't wait for that because the actual patients will not survive to go through that workflow. And this is a very, very standard workflow in the US. I have to apologize. I'm not sure whether it's the similar workflow in Japan, but this, or the times, the workflow should be similar in Japan, but I'm not sure whether the actual timelines are similar. So we thought about it and we asked ourselves, can we engineer something better? Can we actually intervene and what is the earliest point we can intervene? And that earliest point was we have a biopsy. We have something from that tumor. And when you have something from that tumor, you usually get this slide, this H&E slide. And what you can do with it, the slide actually has the stained tissue, is you can actually train an AI model. And what we did in this specific case is to do a multi-resolution learning AI model. We look at, just as a pathologist, at 5x zooming and 20x zooming. We train 2 different independent models. Eventually, we get 2 results, we agglomerate them, and we produce a specific outcome. Now, we wanted to test that and whether this worked for a biomarker. And we wanted to go for a biomarker that's established and well-known, and we focus on something called homologous recombination deficiency. And the reason homologous recombination deficiency is useful is because it's, first of all, an FDA-approved biomarker in multiple solid tumor indications, but second of all, we know what to do. We know how to treat these tumors. We give them platinum therapy and PARP inhibitors because there is synthetic lethality. So if you do not have homologous recombination deficiency, so if you're HR proficient and you give people these treatments, well, the cells live very happily after, and nothing happens. But if the cell, specifically the tumor cells, has this deficiency and you give them the PARP inhibitors, they die. So you're targeting the cells that have this deficiency, and you can do, well, the pure definition of precision oncology. You're targeting what's in the tumor. In the United States, HRD has been approved in at least 4 different indications. There is a lot of details of how the approval has gone and exactly what treatment can be given, but essentially that's breast cancer, ovarian, prostate, and pancreas. And our focus was on breast and ovarian because we thought that's the place where we can have, we can intervene and when there is large patient populations. Now, what we did is we took data and we performed our training using the Cancer Genome Atlas data, so a publicly available data set. And again, once you have the training, and the training uses the genomics and the H&E slide, you don't need molecular profiling anymore. You need just the digital image of that slide. So you take the digital image of that slide, you run a model, and you get a result, yes or no. Is this patient HRD positive or is it not? And the result is instantaneous. It gets there into seconds. And then an oncologist can make a decision whether to treat if the result is positive, which would be following the NCCN or the National Cancer Center Network's guidelines, or whether not to treat and do something else. Now, we wanted to evaluate our model, and the first thing we did is we compared it with the other FDA-approved biomarkers. And there's many ways to compare that, and one way to do it is area under the curve, and we got an area under the curve of 0.8. You can think of it as our accuracy. And we can do that in a held-out set or an independent set. Now, you can see how some of those positive tumors look like. They have a lot of red, so we can find what's wrong with them. And you can see how some of those negative look like. We cannot find any evidence or any morphology phenotype that tells you the tumor have it. An AUC or an accuracy of 80% is disappointing, right? Nobody wants an 80% chance of whether they can treat my cancer or not. But what we realized is that a patient doesn't care how well you match the biomarker. A patient cares how well you treat them. So if we detect the biomarker, is our approach as good as, worse, or better than existing treatments? And that's exactly what we did. We took in this specific case a clinical trial from France with metastatic breast cancers. Every single patient was given platinum therapy. And first we ran our deep HRD test, and what we can see is that 23 patients were positive, 54 were negative, and our test could specifically separate them, and we can predict better performance, and that's when you adjust for age or subtype. And then we ran the standard of care in France, which is the BRCA1, BRCA2 mutations, the key mutations in this gene, or some extended panel that they do. And again, they were kind of capturing it, but they were capturing 9 people with the BRCA status or 13 people with the HRD gene status. So they were missing about half of the population that's going to respond. And then we took these tissues and took the US standard of care, which is Myriad's test, which is the genomic HRD score, which was actually performing quite poorly on these specific samples. And again, we used one additional standard. So by these comparisons, examining this clinical trial, we can actually see that we capture more patients and they respond better. I should say we have replicated that in several other data sets, which I'm not showing here due to time restrictions. Now, this works for breast cancer. Can we transfer the knowledge to ovarian? So we did a transfer learning model. We trained one ovarian cancer model. And again, we tested that on platinum-treated TCGA patients, and we can see exactly the same results. We can see the separations. But in this specific case, we were training on TCGA and we were testing on TCGA. And this is not very, you can do it, you need to be careful, but it's not the best possible practice. So we started hunting for another cohort. And indeed, we were able to find one from Memorial Sloan Kettering in New York. And again, in this specific case, patients were giving neoadjuvant chemotherapy. This was done in a very, very different way. These patients were, the staining was different, the scanners were different. So every single possible component we can think of was different. And that's great because you want a robust model that works on different parameters. And again, we ran our deep HRD, and you can see the separation here. And then we ran what MSK has as part of their platform. One is the BRCA1, BRCA2 status. Again, kind of gives you a good result, but misses about 75% of the patients. And then again, you can put the extended type of panels that they use, and that's going to miss about a half of the patients who respond. So not only we were able to show that we capture more patients, but again, we can see that they're responding just as good as the one that are being missed by the other molecular tests. So how does it work? The big problem with AI is black box AI, something that tells you something that works, but you have no idea why it works. And there are very bad examples for that. There are AI models recognizing dogs from wolves, and then people realize that the wolves, the photos are in the wild, and for the dogs, the photos were at home. So it was actually recognizing the background, it was not recognizing the animals. And we wanted to figure out why does the model work? And essentially, what it was seeing is more neoplastic tissues in the HRD positive cases and less stromal tissues. But perhaps one of the most predictive phenotype here was the presence of macrophages. The HRD tissues, and I suspect I haven't presented the best possible way, the HRD tissues had substantially larger number of macrophages and larger number of necrotic tissues. And that brought about 90% of the prediction power. And it was very powerful because there's prior biological evidence that there are more macrophages in these cases, and there is more necrotic tissues. Pathologists are just not trained to see it by eye, but the AI detects it because it sees the differences. It looks for patterns that the human eye may not capture that easily. Now, we can go back to our workflow and ask the question, can we have an engineering solution where we intervene in the care of patients? And now, instead of after the patient biopsy is there, we don't need the rest of the pathway. We can run an AI model, and that AI model can give you a negative result or a positive result. As a matter of fact, you can run multiple AI models for multiple biomarkers. So when the pathologist writes a report, you actually get a list of AI biomarkers, whether they're positive or whether they're negative. And again, that tissue should probably go immediately after that for genomic sequencing, but the treatment can continue while the rest of the workflow works. And in the US, being very decentralized, there's 186,000 clear certified laboratories. Some of them have scanners, some of them don't. The major centers do. But in systems like Japan, in systems like Europe, where there is centralized healthcare, digital slides are actually quite common. And this allows to utilize them for making clinical decisions or pre-screening, and to either making, you can have an oncologist either made an immediate decision, or actually you can reduce the number of patients that are being sent for molecular diagnostics. Now, again, we've thought a lot about accessibility as part of this project. And one of the things we thought we'll be doing is to try to deploy this in developing countries. Unfortunately, that funding got slightly cut. But what we wanted to do is to see, well, okay, it works in countries that have high economic development, but what if you want to go in Africa? What if you go, I'm from Bulgaria, what if you want to go in Eastern Europe, where you do not have this infrastructure? We still just need the image, but they cannot afford to give 500,000, 200,000, 100,000 for a scanner for a hospital. Just this money do not exist. So one of the things we were very excited is, well, they all do H&E slides. And again, in the US, it's 3 to 6 weeks. In Bulgaria, it's months because they send any profiling to Switzerland and then they return it back, and it takes forever for a number of custom reasons also. So you have places that you can really make a difference. And what we actually figured out is you can take the image very easily from a cell phone. You need to buy a very small device that allows you to zoom in, and you can take that image and you can just use your point of care, or you can use AI attach to detect the biomarker and to see it. And again, you may ask yourself, well, wait a second, okay, they may not, if they don't have the detection, if they don't have the scanners, will they have the treatment for these specific biomarkers? And the answer is they will not have PARP inhibitors, which is one type of treatment for HRD, and it costs about $60,000 per month in the US. But they will have platinum therapy, which is the other type of treatment, which is cheaper, which is in all healthcare systems. And they can prioritize to give the cheaper treatment now at this moment so patients can respond better. So that was our idea, to make it more accessible, and we had a prototype that was somewhat working. And again, all of this was summarized in the Journal of Clinical Oncology paper, showing how it works, showing how we detected, and showing comparisons with different FDA-approved biomarkers. So just to summarize my talk, in the first part, in the second part, I showed you genome analysis for precision oncologists, where we focused on intervening at the pain points to enable faster, more accurate, and more cost-effective detection of clinically actionable biomarkers. I showed you HRD. We have 3 or 4 other biomarkers in the pipeline. We think that this can make a real difference. The second thing that I showed you in the first part of the talk was trying to understand what causes cancer. And there, the combination of AI, machine learning, and large-scale genomics give a very, very good pathway towards preventing what causes cancer, towards potentially eliminating, or at the very least managing better, the biggest killer in young adults, which is the early onset colorectal cancer. And again, I showed you 2 applications. There are many additional discoveries and applications that are being expected. I like to think as an AI as a fancy calculator. We can calculate much faster, much more, much better. And with that, we can actually do more, faster, cheaper, and better. And whether it's for prevention, whether it's for diagnosis, whether it's for treatment or drug design, I think there is a huge potential of changing the whole clinical workflow, especially when it comes to oncology. And with that, thank you so much for your time, and I'm happy to answer any questions you may have.
Thank you, Professor Alexandrov. For those in the venue, if you have any questions, please raise your hand. Questions in Japanese are welcome. 会場からの質問は日本語でも結構です。どうぞ挙手をお願いします。どうぞ。
Ludmila, good talk. Thank you very much. And then the simple question would be, you found the signature for the HRD response, right? Predict the due to pathology. But actually, did you check whether that is correlated with the response to the PARP inhibitors?
Yeah, so it's a great question. Did we check with PARP inhibitor? So we have checked on the real-world data sets. And real-world data sets are data sets where patients come to the clinic and you give them treatment, but they're not controlled. What we're doing at the moment is to take patients from clinical trials, the ones that were actually approved, the PARP inhibitors, and to see, can we replicate the results? So we're trying to get this data, and we'll do that. But in the real world, the answer is yes.
Thank you very much. Thank you very much. So I'm question about HRD diagnosis. Obviously, HRD diagnosis is probably dependent on molecular profiling, signature analysis, structure variation, other stuff, and so. So on the other hand, AI can do anything. We have the data of the true data and of the test data. So what's kind of data you're using about the true HRD diagnosis?
It's an absolutely great question, because that's one of the biggest problems. This is one of the things I didn't get to. This terrible biomarker. In the US, there is about 33 different definitions of the biomarker. and they do not agree with themselves. And in fact, there is 2 FDA approvals, and the agreement is 80% between them. So what's the ground truth? It's a very hard question. I think most people would agree that the person who has germline BRCA mutations with a second somatic hit is definitely HRD. And people who have very, very stable genomes are definitely HRP. But there is a whole spectrum, which is probably 60 to 70% of all the other patients in between. So the way we trained our models is to take the ones that are very much, we know are positive, and the ones that we know very much are negative. And essentially, we did soft labeling with scores. And then our comparison, our argument, at least my argument is, we don't definition. We care what works in the clinical trial. So if we take that and train on something that you can enrich for positives and negatives, and you can get a better response in a trial, that's what I think we want.
会場からもう一つだけどうぞ。あと2つでございます。どうぞ。あと2つなのでいただきましょう。オンラインの方は日本のではなくて英語で Q&A を使って
Thank you very much. Very exciting, talk. And just want to understand the AI, the future of the AI from the regulatory perspective. Actually, I'm using the cloud code by myself. I pay $200 per month, and it's up to me to pay. But will the healthcare, for example, in Japan, it's a national healthcare system. So I don't think, MHLW will pay for that. It's very hard to get, the approval, AI-based, diagnosis or whatever. So how do you proceed? How should we overcome this? Because, just as Dean mentioned, we need to bring it into the practice. Otherwise it's just for fun.
It's an amazing question. I think the next talk we touch a bit about that in the US. I should say for AI, and again, it was in my disclosures, we have a company where we're trying to commercialize this specific technology. And the AI regulatory market is, to me, the Wild West. Everything is happening, and the regulators are trying to catch up with the technology and trying to understand what to do and how to do it. Same with the reimbursement, same with the payers in the US system. I think AI has one huge advantage, and that huge advantage is it can be much cheaper and much better. And that, I'm showing you some examples here, but there's examples for drug discovery, there's examples for treatments. So that is going to be a huge initiative, whether for systems like the US, where you have the insurance company wanting to pay less, or national healthcare systems like Japan or the European style, where the government wants to pay less. So I think it will move forward, but I suspect the next decade will be turbulent, and there will be many, many changes.
では最後会場からもう一問だけいただきます。
Thank you for your talk. Earlier you mentioned that there were unknown mutagenic signatures. We didn't know what the cause of those signatures are yet. And I was wondering if you think there are any good ways to identify those.
I do. That's what we have a whole new grand challenge that started, which we're spending the next 5 years doing it. I'm not sure whether our approach will work, but we do have. And the simple idea there is we are going to focus on the biochemistry, the biochemical modification, the adducts. And people may not appreciate that, but we know a lot about mutations, changes in the genetic code. But the biochemistry, the biochemical modification of bases that lead to these mutations are a bit of the dark matter of what's happening. And that's where our focus is. I don't know if it would work, but it's at least an avenue that we're taking.
Thank you.
Thank you, Professor Andrew.
Thank you so much.
We may pick some questions online at the panel discussion session. And next, we will have Dr. Hidewaki Nakagawa, Team Director of RIKEN. Dr. Nakagawa, please.
Okay, thank you very much for inviting such wonderful sessions, and also the actuaries of Vladimir recommended me. It's a pleasure. 日本語でいきますね。OK。Let's talk to Japanese. はい。これはポイントはない。これかね。はいはい。はいはいはい。はい。今日お話ししたのお題としては、イムノジェノミクスと AI ということで、僕もがんゲノムの方をラドミンの方と一緒に、ずっと国際ICGC プロジェクトですね。でずっとやってきております。特に障害はございません。で、プレシジョンオンコロジーというキーワードで考えた場合に、このようなワークフロー、クリニカルなレベルではこういうふうなワークフローで今現在行われております。プレベンション、ダイアグノシス、それからトリートメント、モニタリングという形になって、それぞれのステージで、いろんなこのゲノム情報ですね、が使われてきております。先ほどあったようなプレベンションということになると、リスクアセスメントっていうところで、ゲノム情報が使われる。それから診断ということになると、先ほどの HE の像もそうですけども、そういうトゥーマーのクラシフィケーションっていうのに使われますし、それから今はプレディクションですね、トリートメントのレスポンス、治療のレスポンスを予測するというのがマーカーとして使われてきております。それから実際の治療としては、さまざまな今治療があって、モレキュラーセラピーについては、やはりそういうふうなゲノム情報っていうのが使われてきております。それから今回の題としては、しますターゲットアイデンティフィケーションでね、シーズ探索というものについてもゲノム情報が今現在使われております。それから最終的に治療が終わった後にモニタリングを行いますけども、現在そのような再発の予測というので MRD というふうなシステムが使われておりますし、それから薬の副作用を予測する PGX というふうなフィールドもございます。で今日お話しさせていただき、いろんな情報がゲノム情報がこういうふうながんの臨床で使われてきておりますけれども、今日お話しさせてもらうこのテューマークラシフィケーションとプレディクションと、あとはそのシーズ探索というものについてお話しさせていただきます。これは1枚だけのスライドになりますけれども、最近我々がやってきました腎がんの診断ということになります。腎がんというのは、非常に複雑というか病理学的にこのように20種類ぐらいの組織系があって、これを診断するっていうのはなかなか難しい。日本の中のトップの病理の先生と話しても非常に難しくて、このようにアンクラシファイド RCC という形で診断ができないような腎がんというものも存在しております。で、これのサンプルをRNA シークエンスですね。RNA のプロファイリングを行いますと、このような形できれいにこういうふうなクラスができるようになってきておりまして、で特にこのアンダイアロシス、診断不能腎がんというものもこのように特定の腎がんにきれいに分布されるということが分かってきております。このような形でRNA 発現情報を使って腎がんの診断の補助ができないかというのを現在考えております。これが事業化できないかというので、現在考えてきております。ダイアグノシスについてはこれで一つになります。で、まず一つのメインのタイトルとしては、そのがん患者さんのクリニカルアウトカムを予測できないかと、ゲノム情報、オミクス情報、AI を使ってできないかということについて、現在取り組んできております。で、これまではシングルバイオマーカー、それからマルティブルマイオマーカー、さらには、もっとコンプレヘンシブな全ゲノム、それから全 RNA の発現を使って予測できないか。それからDNA を使うのか、RNA を使うのか、プロテイン、それからメタボライト、先ほどラドミーが話しましたね、イメージングですね。ラディオミックス、それから HE のデジタルパソロジーを使って、それからそれをクリニカルインフォメーションと使ってできないか。さらにはそういうふうなサンプルですね。がんのサンプルを取るのには、インベーシブな方法とノンインベーシブな方法でやる形ので、 こういう非侵襲的な方法でできるかどうか。それからもちろんそのアルゴリズムですね。AI を使ったアルゴリズムの正確性、それから再現性というものも議論になってきます。それからあと、先ほどありましたけども、実際にこのバイオロジーとして見つかったマーカーなり、見つかったアルゴリズムが本当にこのバイオロジーとして正しいのかどうかというものの議論になってきます。で、我々やってきているのは今このキャンサーティッシュビフォートリートメントという形ですけれども、治療前のサンプルですね、バイオプシーサンプル、特に消化管の場合は、日本では簡単に胃カメラ大腸ファイバーをやって、この腫瘍のサンプルを取ることができますので、非侵襲的にサンプルが取ることができます。このサンプルから DNA、RNA の解析、それから免疫の解析、プロテオミクスの解析、もちろんあとクリニカルインフォメーション、それから先ほどのあったようにデジタルパソロジーだとか、レディオミクスだとか、イメージングのデータを使って、これを機種法にかけて、これも一つ話題になってきます、エクスプレイナブルな AI ですね。やっぱり臨床の実装ということになったとか、あとレギュレーションを考えた場合に、実際に何が本当にこの AI の中でどういうファクターが重要なのかということは少なくとも示さないとなかなか理解が得られないと。こういう情報を使って患者さんのレスポンスだとかアウトカムを予測するということを行っております。でこれ実際の食道がんの例です。121例の食道がんですね、扁平上皮がんのネオアジュバンドケモセラピー、これ普通に行われているような治療前のサンプルを使って、バイオプシーサンプルを使って DNA、RNA の解析を行いました。DNA の解析から DNA のホールゲノムシークエンス、それから RNA の RNA シークエンスのデータからさまざまなパラメータを抽出してきております。でこれはプレリミナリーなこのプロジェクトを行われる前にフルホールゲノムシークエンスをやって、実際に変異のパターンとこういうふうな化学療法の感受性というのが関連があるかどうかというのを見てみたんですけども、以前はこの P53 の変異ですね。P53 との変異が何か関係あるんじゃないか、DNA の修復とは非常に関係しますので関係するんじゃないかということを言われてましたけども、こういうことは分かりませんでした。ですけども、他の例えば先ほどラドミルが話したように SNB のシグニチャー SBS92 というものと非常にこの統計的にレスポンダーとノンレスポンダーとが関連すると。これは92っていうのはタバコのスモーキングシグニチャーですね。タバコの関連のシグニチャーになってきます。それからコピーナンバーですね。食道がんはP53 の変異が多いです、ほとんどですけども、さらにはコピーナンバー異常っていうのが非常に起きております。エクステンシブに起きてきております。で、レスポンダーとノンレスポンダーとの比較をしたときに、9番染色体だとか、12級の染色体の異常っていうものが差があると。で、その12番級の染色体のそのデリーションの部位を見てみると、このネガティブレギュレーターオブ NF-κB シグナリング、炎症系のシグナルの関係の遺伝子がこの部位に載ってるから、もしかしたら炎症と関係あるかなというふうなことを考えておりました。でさらに RNA のデータから、さらにこのジーンセットエンリッチメントですね、DNA の発現で、レスポンダーとノンレスポンダーの比較をした場合に、このインターフェロンガンマのレスポンスに関連する遺伝子というものがエンリッチしてきているということが、レスポンダーの方にエンリッチしてきているということが分かってきております。で、さらにコピーナンバーの今度はシグニチャーですね。先ほどラウミがありましたように、このコピーナンバーのパターン的解析をUK のグループと一緒にやってきております。その中で、この、シグニチャー6っていうシグニチャーですね、パターンですね、が、この関係しているということが分かってきておりまして、で、実はこの論文にありますように、同様の卵巣がんについても、このようなシスプラチンベースのケモセラピーと、このコピーナンバーシグニチャー6っていうものが関係しているということが分かってきてました。それから RNA のデータから 免疫のシグニチャーっていうものも予測エスティメートですね、評価することもできるようになってきております。で、RNA のデータからこのように、高中球ですね、高中球 CD4 ポジティブの T 細胞、B 細胞、それから CD8 ポジティブの T 細胞が、多いがんというものの分類ができてきておりまして、このような分類を行った後に、実際に、化学療法との感受性を見た場合に、このニュートロフィルグループ、ニュートロフィルが多いグループについては、化学療法の感受性が悪いと、低いということが分かっていました。こういうふうないろんな情報ですね、マルチオミックスデータ、結局81個のパラメータが関連ということでしたので、この80個のパラメータを先ほど言いましたエクスプレーナブルな AI であるマシンラーニングであるランダムフォレスト法を用いて、こちらの研究員の翔太笹川が解析を行って、このような5つ6つのファクターが重要であるということが分かってきておりました。で、これが最終的なディシジョンツリーですね。ランダムフォレストの場合は、マシンラーニングのランダムフォレストの場合は、このようにディシジョンツリーというものが、レスポンダー、ノンレスポンダーの予測という時に、どういうファクターがどれぐらいの形で効いているかっていうのが、一応可視化できるというのが一つ優位なポイントになってきます。このように、例えばクロモゾームの12番のところのあるかなしかによってこういうふうな形が分かれてきまして、さまざまなファクターを組み合わせることによってこのように、ある程度のこの層別化というもの、化学療法のレスポンスの層別化というものができるようになってきました。でこれが実際のサンプル、数は少ないですけどもサンプルですけどもこのような形でレスポンダーとノンレスポンダーのある程度の層別化が出てきて AUC としては大体 0.8 ぐらいは達成できるというふうな結果でした。で、もう一つ問題なのは、これは先ほどのラーニングセットになりますので、別のデータセット、テストセータデータというものを32サンプルを持ってきまして、同様のアルゴリズムで解析した場合に、やはり大体 AUC としては 0.8 ぐらいは達成できるだろうということで、このような再現性の問題というのも、ある程度は解決できているのかなと思います。で、もう一つはそのバイオロジーですね。最初言いましたね、バイオロジー。そしたら先ほど言いましたように、高中級が関連しているということが分かってきましたので、高中級のマウスモデルをちょっと作ってみてやってみました。で、人の場合はできないですけども、マウスモデルで、食道がんの扁平上皮がんのマウスモデルを作って、化学療法シスプラチンとそれからニュートロフィルのディプリーションをかけるような操作をして実際にニュートロディプリーションをかけるとあとシスプラチンを併用することによってこのように化学療法の効果っていうものが良くなると使用が抑制されるということが分かってきておりまして、バイオロジーとしてもリーズナブルな結果かなと思います。で、今度は別の胃がんですね。胃がんでも同様な研究をやっていました。これはがんセンターとの共同研究になりますけども、これも化学療法、ネオアジュバンド化学療法ですね、をやった場合に大体走行率は 46% ぐらいなんですけども、これの DNA、バイオプシーサンプルを用いて DNA、RNA の解析を行って、さまざまなパラメータを抽出してきました。で、ちょっと字が小さくて分かりにくいですけども、これは例えば、レスポンダーとノンレスポンダーですけども、インターフェロンガンマですね。インターフェロンガンマだとか、インフラマトリーレスポンス、それから IL6、JAK3 パスウェイ、それからイムノスコアっていうものが、一応統計的にレスポンダーとノンレスポンダーの差があるということが分かってきております。で、また先ほど同様に、高中級などのこういうふうな分割をRNA のデータから評 評価して、このような分類を行って、実際にこれらが化学療法のレスポンスと関係あるかどうかというのの解析を行いました。先ほどの、どういうふうな機械学習法のモデルがいいのかというのも、先ほどの笹川の方でいろいろ検討してきまして、内部ベイズ、それからランダムフォレスト、サポートベクターマシンという3つのアルゴリズムで、解析しましたけども、やはりランダムフォレストの方が一番分かりやすくて、かつバリューも高いということで、同様にランダムフォレスト法を用いてのマシンラーニングを用いて、同様な解析を行ってきました。それからどのファクターが重要なのかというのも、こういうふうなインポータンスで、右端のこの辺のファクターっていうものが重要だろうということで、その中には B セルだとか、先ほどのニュートロフィルだとか、コピーナンバーのシグニチャーだとかというものが、この化学療法のレスポンスと関連しているんではないかということで、これらの情報を用いて、ランダムフォレストで解析を行って、同様にディシジョンツリーを書いてみると、最初に上がってきたのがニュートロフィルのシグニチャーになってきて、他にもいろいろな化学療法だと、インフラマトリーレスポンスだとか、あと CD4 の T 細胞のシグニチャーだとかっていうものが関連してて、そういうのを組み合わせながらこのような形でディシジョンツリーを書くことができました。でこれ実際にこれも数が少ないから難しいところかもしれませんけど、 AUC は 0.8 ぐらいで、やはり大事なのはバリデーションデータテストデータということで、他にサンプルがなかったので友人の MD アンダーソンの先生にちょっとお願いして、同様な研究の論文が出てましたので、そのデータを使って同様に AUC が0.7 ぐらいは一応できるということがバリデーションすることができました。ということでなんですけども、バイオロジストとして少しちょっと面白かったのが、食道がんの場合と胃がんの場合で、このニュートロフィルですね、高中級のシグニチャーが逆方向に効いていると。化学療法に対する効果としては逆方向に効いているっていうことが分かってきまして、でちょっとそのバイオロジーとして今現在このニュートロフィルについて注目しております。でニュートロフィル、テューマーアソシエイトニュートロフィルという形で、このようにさまざまな実はファンクションを持ってきている。でがんについてもテューマーオンコジェニックテューマーグロースに効いているものがあったりだとか、テューマーサプレッシブに効いているものがあったりだとか、また免疫にももちろん関わってきてますし、いろんなファクターを持ってきているということが分かってきておりました。ということで、現在アドバンスのイムノジェノミクスとしては、現在今このシングルセル解析というこのニュートロフィルにフォーカスしたニュートロフィルとか免疫細胞にフォーカスしたシングルセル解析を使って、それを AI を用いて解析しているということを現在行ってきております。これはワークフローになりますけども、組織からシングルセルの組織を手術で取ってきた細胞をシングルセルのディソシエーションをかけて、一応それを保存しておくと。で保存してきた場合に、それを起こしてきた場合に、起こしてきて、免疫細胞が起きてきますので、それを現在の一番のシングルセル解析を行って、どういうふうな細胞がいるのか、免疫細胞がいているのかということをやってきております。その中でも、一番問題なのは、この辺の、しますね。この辺の細胞のアノテーションですね。アノテーションがなかなかまだ難しいということなんですけども、これを現在今もう AI を用いて、まさにこの RNA の発現データを用いて、どの細胞かっていうふうなこのはある程度かそこそこのアノテーションができるようにはなってきております。でこれは実際にどういうふうなサンプルで、先ほど言いましたように凍結保存してきたやつをさらに起こしてくるというふうなところですので、どういうふうなちゃんとそこでバイアスがかかってないかどうかっていうのを評価したのが、近畿大学の柿見先生のサンプルを使って、実際にファックスで、シングルセルディソシエーション後にファックスをかけたやつと、 フローズンして、それから起こしてきた免疫細胞が違うのかどうかっていうのを評価したものになりますけれども、基本的にコンポーネントとしてはもうほとんど変わらないということが分かってきております。で、実際に、食道がん、胃がんの組織からシングルセルイムノセル解析ですね、行ってきて、約10万個の免疫細胞ですね、を抽出してきて、このような形で AI を用いて、その細胞のアノテーションというものを行ってきました。で、なんですけども、実は先ほどもニュートロフィルに着目していると言いましたけれども、実はこのプラットフォームでニュートロフィルの解析はできないということが、当時分かってきていました。で、これを会社の方にいろいろ問い合わせしていまして、実際に同じような同様の解析をしている仲間ですね、といろいろ相談しまして、それ用のAI なり解析の手法っていうのを開発して、なんとかこの免疫細胞です。好中球をうまくアノテーションできるような手法っていうものを開発してきまして、約10万個の免疫細胞、好中球っていうものを抽出することができました。で、これを AI で分析してみますと、このような好中球であってもいろんな好中球がこういうふうな形でいけると。で、それをアノテーションしていますと、アンティテューマーであったりだとか、それからプロテューマーだったとか、あるいはそのバイオロジーとしてはネクトーシスというに関連している好中球だとか、さまざまな好中球にこう分類できると。で、これは胃がんと食道がんでやっぱりこのアンティテューマーの端とプロテューマーの好中球が違うんだっていうことが分かってきましたので、そのデータですね、その好中球の発現データを用いてシグニチャーをさらに作成して、で、今度はバルクのデータですね。バルクのデータで、実際にこのシグニチャーっていうのが検出できるかというものをやっていきました。これは TCGA のデータになりますけれども、TCGA のデータを使っても、やはりアンティテューマーのニュートロフィルが多いサンプルについては、予後が良かったり悪かったりだというふうなことが分かってきております。ということで、こういう形でシングルセルの解析ですね。で、いろいろこういうふうな主要のストラティフィケーションだとかクラシフィケーションができることが分かってきておりましたので、今現在は次のステップとしては、このやっぱりドラッグデベロップメント、特にシーズ探索にこういうのが使えないかということに今考えてきております。で、さらにこの発展系としては次のウィアンの方で発表があると思いますけれども、我々肝臓がんを主にやってきましたので、肝臓がんについては今現在免疫療法っていうのがもうファーストラインになってきておりまして、もうがんとしては他のがんもそうだと思いますけれども、免疫の病気であるということが分かってきております。なので先ほどの同様の免疫解析とイムノジェノミクスの解析をつかないと新しいシーズ探索ができないということが分かってきておりますので、今現在はこれは今度スペシャルトランスクリプトム解析ですねを導入して、でそれをさらに AI を用いてシーズ探索を行ってきております。これは54例のバイオプシーサンプルですね。肝臓のバイオプシーサンプル、ICI 投与前のバイオプシーサンプルを用いて、で、それの実際の ICI を投与した後に、効いたか効いてないかというものの解析を行ってきました。こんな感じで、スペシャルトランスクリプトの解析をすると、非常に汚いデータですけども、それをなんとかマージすることができて、でその中で出てきている細胞のパスウェイ解析だとか、セルセルインタラクションの解析と、その化学療法のレスポンスの関係というものを解析しております。で、これ一つの解析の手法なんですけども、セルチャット解析といいまして、実際に細胞ですね、いろんな細胞が混じってますので、それぞれの細胞とのインタラクション、ライガンドとレセプターの関係、発現の情報をひっつけて、でどの細胞でどういうふうなセルセルインタラクションが起きているかと。で、それのレスポンダーとノンレスポンダーで、どういうパスウェイがエンリッチしているかどうかというのが見たことになります。そうすると、レスポンダーの、ノンレスポンダーに一番トップに上がってきたのは、実はウィントシグナルパスウェイということが分かってきました。これはちょっと我々もちょっと 当たり前といえば当たり前かもしれないですけど、ウィントシグナルパスウェイですので、ベータカテニを関わるウィントシグナルパスウェイは、がんの世界では、イムノサプレッサーと言われてて、かなりそういう免疫に対してのレジスタンスを創設するというものなパスウェイになってきます。そういうものがちゃんと上がってきているということが分かってきまして、この下の他のパスウェイっていうものも、もしかしたらこういうふうなレジスタンスですね、ICI に対するレジスタンスと関連している可能性があるということが分かってきております。こういうものがシーズンになるかなとは思っております。で、もう一つの方法としては CMAP 解析といいまして、RNA のシークエンスデータがノンレスポンダーとレスポンダーを比較すると。で、それの中で、ディファレンシャルエクスプレッソジーンというのを抽出して、そのパターンと先ほど言ってたシグニチャーとこの CMAP のシグニチャーとの比較を行うということをやってきております。で、この CMAP っていうのは、これはもう昔からアメリカの、ブロードインシチューツの方で作ってきたデータで公開されているデータでして、50種類から70種類の細胞株に、数万、今確か数万種類ぐらいのコンパウンドまたはジェネティックリエージェントをかけて、その発現パターンを、その発現の推移ですね、をこう見てきているようなデータになります。で、そのデータと実際に臨床サンプルのデータ等を比較して、コネティブスコアとネガティブスコアですね、つまり、薬をかけることによってその発現パターンが変化すると同じようなものが薬がないかどうかというのを探索するような技術になります。このような感じでドラッグシグニチャーとそれからディジーズシグニチャーですねが反比例するようなそういう薬がないかどうかというのを探索するような形になります。で、これを実際に先ほどの、まだスモールスケールですけども、肝臓がんの ICI のレスポンダーとノンレスポンダーをやって、で、ノンレスポンダーですね、レジスタンスのところに、と反比例するような薬がないかどうか。で、細胞株は HEPG2 の細胞株、肝臓がん細胞株を用いてのデータを用いて探索をしていました。で、このような形ですると、HEPG2 の細胞ではいくつか優位な、統計的な優位なドラッグっていうものが引っかかってきています。反比例するですね、シグニチャーが反比例するようなシグニチャーが低まってきまして、これ書いてますけども、いろんなその免疫関連のモジュレートする薬だとか、あとは実際にクリニカルトライアルやられている薬だとか、いくつかこういうふうな有望な薬が上がってきておりまして、これはあくまでもインフォマティクスによる探索ですので、もっと必要なのはこれをバイオロジカルにバリデーするということがもうなってきております。ということなんですけども、我々はなかなかまだそこまでは手出せないというので、もしご興味のある製薬の方がいらっしゃいましたら、またお声をかけていただければなと思います。最後のスライドになります。最初のスライド、これまでの大きなスライドのデータは、こちらの研究員の笹川がやってきたものであります。それから、近畿大学の方は食道がん、それから広島大学は胃がん、肝がんです。ナショナルがんセンターの方は、山田先生の方からは胃がんのサンプルをもらって、MD アンダーソンの反乱だとか、あと ST 解析については今、柏の鈴木先生と一緒にやってきております。以上になります。どうもありがとうございました。
Thank you, Dr. Nakagawa. Let's move on to the next presentation. We will have Dr. Wong Yan Park, MD, PhD, Chair of GXD. As he leaves the venue after his presentation, we will have a Q&A session just after his presentation. So for those attending online, please submit your questions in the Q&A box during his presentation. Dr. Park, please.
Okay, thank you for the kind introduction. Today I'm gonna introduce about how we can use the spatial omics data for drug development. So our company be a GXD be genome multiplied by data. So especially that we want to combine the genome data and then clinical data together for industrial, the development purpose. So we are located in the LINK lab in Kashiwanhwa. So we have some strong, in the beginning, actually, we are closely connected with the, communicate with the LINK lab. And then, yeah. So it's my disclosure. And also I have two parts, like the how we can use the spatial omics data for your clinical activity. And also we can find, discover some potential target for new drug target, new drug development. Also, I want to raise our strategy to how do we use the spatial omics data for target development. So we founded the company in Japan in 2023 to work with the Scrum Monster project that be organized by NCC and Dr. Yoshino. And then he running this Monster project is simply we can say that it is a kind of prospective quote with the multiple IIT. So maybe utilize that genome data and also spatial omics data, other omics data to find that some indication for some IIT. And so try to find some new indication for the treatment in the cancer patient. So he combined the Monster project combined the omics data plus in the strong IT back support. Also, based on it's a clinical trial level control the data and also other process. So directly utilize that information for real world data also for the approval in the PMDA in Japan. And also, they invite the multiple scientific researcher in the world widely. So maybe you can join this project to utilize that data and also to discover new the knowledge out of this project. So we are running this project as a participant, and also we are taking part in the spatial omics and other omics data generation. So, for example, I briefly introduced this project based on genome profile and spatial omics profile. We can assign the patient for specific IIT. So IIT means we design, and then a participant can design in this study, and then we load the patient. Approximately will be enrolled about 3,000 patients. So we can see the outcome of the patient based on our biomarker, and then how we can improve the patient outcome based on this clinical trial. And then also we can use the real-world data. We already have in NCC in Japan, and then we use that kind of information with for example, HER2-positive cases. And then they have a lot of the HER2-positive colon cancer data. And then if we run HER2-positive, like HER2 in the colon cancer, probably can compare with the other data set together. So this project, Monster project, is starting from the 2015. And then in the beginning, they used just cancer genome panel, but they evolved continuously to use at this moment, they use the spatial omics data, plus Olink proteome data and other omics data, including the microbiome. And they also incorporate the MRDs, the test in this project. So today, this is the ongoing project, so I cannot conclude that or maybe present anything about this project. But actually, I can show an example operated by GXD that be understanding the MOA of the neoadjuvant chemotherapy in breast cancer. So because the neoadjuvant chemotherapy is the routine. The process treatment for the breast cancer, but actually we want to know the response, the mechanism or response mechanism to improve that outcome, also to find any potential new drug targets. So our study we use the post-treatment sample. The neoadjuvant chemotherapy means once we found the breast cancer, and then we start to treat before the surgical section, we treat the patient based on the subtypes. In this case, actually, we focus on the TNBC, triple negative breast cancer, and then treat the patient, and then with the chemotherapy, and then after the period of time, and then they before they start the surgical section. Sorry, actually, for surgical section, we can find that there's no tumor left, or maybe some patients have still some the tumor cells left. So we can say that there is a partial complete response or a partial pathological response, and then we can say this tumor left on the tumor, breast tissue, and then after the surgical resection, we can wait to see the outcome of the patient. So even for neoadjuvant chemotherapy and surgical resection, some patients have the relapse after the 2-year or 5-year follow-up. So we can divide on 2 patients, like after the neoadjuvant chemotherapy and then surgical resection. Some patients have the no recurrence, and then some of them have some relapse in the tumor in the breast cancer. So we divide into two groups based on the response to the outcome. So the relapse cases, we say the dismal cases, and then this is the no relapse in within 5 years. That we can say that non-relapse, non-dismal cases. So because we have the surgical samples, and then also we can follow up the 5 years. That means we can use some sample for 5-year-old PARP sections and for the spatial omics analysis. And then we use the 16 cases, the small number, but actually we can acquire more than 100,000 cells out of the spatial omics analysis. And because we use the post-treatment samples, we cannot make a predictive model, but actually we can understand the mechanism of the action for the neoadjuvant chemotherapy in breast cancer. So out of the spatial omics analysis, we can initially start with the cell type annotation. To identify which cell type present in the patient. And also we want to check that whether the cell type proportion is different between dismal and non-dismal cases. That means I want to know some specific cell types are important determining the clinical response to the neoadjuvant chemotherapy. But most of the genome analysis, we started from the RNA-seq because it's much easier, and also we can use the whole area for the tumor tissue. So, but actually, then out of those just RNA-seq using bulk tissue, we can identify specific signal, but actually we do see some average of the cell types, average of the tumor signature. So it is not easy to identify. But in single cell sequencing or spatial omics analysis, we can separate the multiple cell types to see any difference between dismal and non-dismal cases. That means we can see some specific cell types important determining that clinical response. But out of this figure, we cannot see any statistical significant cell type difference. So our hypothesis would be not hypothesis. Actually, we want to focus on the myeloid cell because the T cell is important, but actually hypothesis will be we want to find some regulator for the immune response in the controlling inflammatory response, and then innate immune response. That may be important for the regulating T cell response ultimately. So when you dissect the myeloid cell into 8 different cell types, and then you can see some cell types showing high and low something, but actually no statistical significance. But out of those multiple cell types, we do see macrophage, CXL line positive macrophage, a little bit slight difference between dismal and non-dismal cases. The CXL line be IL-8, IL line, that is important for the T cell recruitment. So we can say that is high. That means we can expect more T cell recruitment in this patient. So good process, non-dismal cases, they're showing a high level of the macrophage with CXL line positive. And that cell type is very famous because they are already known. Okay, so actually they known that there's the macrophage subtypes in we can say the M1, M2 or suppressive or inflammatory, something like that. But actually, CXL line positive macrophage originally from the tissue resident macrophage. So they upon some inflammatory signal, they activate it and then they secrete the IL line to recruit the more T cells. The other partner will be M2 macrophage, will be SPP1 positive macrophage. They differentiate from the monocyte. So when you have some inflammation or immune response, that monocyte recruited those inflammatory sites, and then they differentiate to SPP1 positive macrophage. And the previous report be the ratio between these good and bad macrophage. The ratio is important to determine the clinical outcome in multiple cancer types. So we also see that the pattern in the dismal cases, they show the high level of SPP1 macrophage, and the ratio is high in the SPP positive macrophage. So anyway, so we to see they replicate their findings, and then but actually we want to know in detail about what is the meaning of those two different myeloid cell subtypes. So spatially, we can see mapping that SPP1 positive macrophage and then CXL line positive macrophage. But it's not easy to separate that because those so many cells are present in the slide. So we calculate that some mixing score. Mixing score means that which cell type, two cell types are mixed together in close area, like we define within 50 micrometer. That means we can localize about less than 20 or 30 cells inside the 50 micrometer diameter. So, and then we calculate that mixing score, normalized mixing score means sometimes if the cell number is high, probably that will be mixed well, but actually, so we normalize that numbers and then calculate the normalized mixing score. And then we found SPP1 positive macrophage, they are mixed together with the cancer cells more. And then CXL line positive macrophage, they are not mixed well, but actually they are mixed well in the some distant area. So when you calculate the 50 micrometer, we need to see the cancer cell and SPP positive macrophage co-localized. But in 100 micrometer or farther than the 100 micrometer, we can see the CXL line positive macrophage, they are co-localized with the cancer cells. Also, that is different between the dismal and non-dismal cases. That means we can say some clinical meaning of those mixing in the distant area will be important for the non-dismal cases. Also, dismal cases, actually, the difference between two groups, we can see within 50 micrometer. So we can find that the ratio is important, but also in the location and the distribution in the next to the cancer cells is also important. And then we also calculate a different way to see the cell density around the SPP1 positive macrophage. Around the SPP macrophage, the SPP macrophage will be the highest, right? And then, but actually cancer cells are second place. That means most close cell type will be with the SPP positive macrophage as cancer cells. We can see that kind of pattern. And then this pattern is also in the with the correlate with the hypoxic signature. So that is well known, but because of SPP is by hypoxic signal or something like that. And the other way be a CXL line positive macrophage, they are co-localized with the other T cells or the immune cells. So we expect that they are not co-localized the cancer cell, but a little bit farther than the SPP macrophage. And then, but actually it's CXL line positive macrophage are co-localized with the T cells, other immune cells together. And especially for the subtypes, you can see the CD8 positive cells are co-localized with the CXL line positive macrophage, but not for the CD4 T cells. So we expect that the CD8 positive cell, we do see we should check the subtype, but actually we expect that going to be cytotoxic CD8 T cell co-localized with the CXL line positive macrophage. And CD4 T cell, usually they are important for the making a TLS structure, but so it's not, maybe it should not be, they don't need to present close to the macrophage or tumor cells. And also, out of the spatial omics data, maybe you can colocalize the cell types. Also, you can colocalize the molecule. So we calculate that which the cytokine is colocalized with the CD8 positive T cells in the responder. And then also, using different method, we can list that some important, maybe potentially regulate CD8 T cells in the non-dismal cases. We define that CCL5 is a top in the out of the two independent the analysis, and then previously reporting the cancer cell, they said that some auto-regulation of the T cell in the tumor immune, the T cell secrete the CCL5, and then, sorry, actually tumor cells secrete the CCL5, and then they recruit the T cell, and then T cell secrete the interferon gamma, and then macrophage differentiate into 6-line positive macrophage, and then 6-line positive macrophage, they also the secrete this 6-line, and then they recruit the T cell. So that is kind of cycle to initiate the immune response in the tumor. So we define that kind of things happen in the non-dismal cases, good prognosis patient. But the other dismal cases, they do not show that kind of pattern. So just simply out of the cell counting or cell proportion, we cannot see that kind of pattern. But when we considering the location and the distribution with the cancer cells, and then we can see that this kind of finding. So, and then we utilize the single cell data because of spatial omics, the limitation of spatial omics would be the resolution is not so high at this moment. I mean, especially in these cases, we use just 1,000 genes, the Cosmex 1K, the panel. So it's not easy to differentiate the multiple cell types. But now you can probably have the 5K or other 18K. Other current version, we probably can identify more cell types, but in this study, actually, we did not use that. So anyhow, we want to specify which CD8 positive T cell is important for those CCL5, CCR5 signaling. So in the TMBC single cell data, we can find, we define that kind of CCL5 or CCR5 they expressed in the T cells. And also when we classify the subtypes, and then we identify those CCL5, they mostly expressed in the TEMLA and also the cytotoxic T cells, exotic T cells. That means we define that kind of, we cannot check that those subtype in our spatial data, but we can say will be we suspect that those CD8 T cell in dismal the cases, they will be the TEMLA or exotic T cell. That may be fully, I mean, we can explain, I mean, the validate our finding, or maybe can prove, not exactly prove, but actually we can propose that the important cell types in the determining the CCL5, depending the response. So if I summarize the spatial omics finding, we can measure the distance between each cell types, and then mapping is a kind of some calculate that distance and then mapping together. And then as you see the non-dismal cases, good prognosis, they're showing the around the cancer cell, multiple immune cells, myeloid cells, they colocalize together. But the dismal cases, the cancer cell, they are isolated from the other cells. And then just a SPFM positive macrophage co-localized with cancer cells. So we expect that the dismal case bad prognosis, they exclude the immune cells out of the cancer area. So maybe we can expect that there's no tumor immune response. So we want to prove the MOA, but also we want to find any potential target. So I think maybe CCL5, CCR5 axis is important. For example, we can use that kind of pathway or to activate the T cell in TMBC tumors to improve that tumor immune response. And to validate our findings, we can check the multiple parameters that I listed in the previous slide. And then ultimately, we found that CCL5 and then CXL line differentiate the response in the TCA data sets. So we can see, if you use these two signatures, if you say the double positive cases, they're showing the highest, best response in the TCA data sets. And then double negative would be a worst response. So maybe you can use that as a kind of biomarker. But also we can, we also try to validate in the other subtype, breast cancer subtype. Sorry, actually, if I summarize this figure, but actually the other subtype do not show that a pattern, but actually just only our findings can be working in just in the TNBC tumor types. That be the breast cancer, the oncologists, they say that biological is quite different entity. So we can expect that TNBC be a more immune response. And also, so we can expect that CCL5 and or CXL line is working just in the TNBC tumors. Okay, we published the paper and with the other findings together last year. Okay, second topic would be I want to introduce how we can use this kind of data, or maybe how can expertise the spatial omics data for the drug development. So reason why we are challenging that kind of things about drug target development in our company will be most of the company and the big pharma, they focus on the known targets because novel target be a high risk. Also, we need more biological the evidence to use that new targets. But maybe some advancement of the clinical, oncology, we need a new targets and new modality. So we try to find a novel target out of the spatial data because currently you are using the RNA-seq data and other genomic data, but actually you cannot get access to the multiple spatial omics data. So as I showed in the, maybe we can find that CCL5, something like that exists out of our spatial data. So I think maybe you can find the new targets, the mechanism out of spatial omics data. So out of the spatial omics data, we can provide some biological, the evidence for the biological evidence based on the spatial omics data. So we can fill the gap for the resolution to prove that this new target is really working or important in the drug development. So we collect about 16, data for the 16 cancer types, and then more than 2,500 spatial omics data. And then fully the property data means we collect just ADC 3D patient or IO 3D patient. That is the most important for the drug development for the ADC and IO drug development company, right? So, and also we have the other level of data like the genome data and also transcriptome data and other data too. And if I look at in detail, we have the 1,500 case for the genome data, spatial omics data, the high level information, and also 400 cases with the clinical treatment match that for example, we have the 200 cases for the breast cancer, not to treat it, and then some of them the response, some of them non-respond. And also we have the matched for the pre and post samples. So that means the pharma wants to look at the data to find the mechanism, also try to find the new targets. And then also we have the spatial omics data for the protein level validation. So mostly in the RNA, we correlate with the protein level, but still they want to see that the protein is really present, or so maybe that is localized in the membrane. So for example, maybe out of pathology data, maybe you can see, pathologists can see this is cancer, this is normal, and this is adjacent area. Maybe affected a little bit, but actually it should be normal. But traditional, the annotation was if you analyze your spatial omics data, probably you can annotate that all these epithelial cells. So epithelial cell means because it's a cancer, so usually we identify as a carcinoma and also in the cancer cells. But based on our analysis, actually, we can separate these 3 areas based on our profile. And then the meaning of this, the separation for tumor normal BA, because you found that some target out of this area probably will be initiate some off-target effect, kind of toxicity. So if I propose maybe if we traditional method, including everything, probably you can reduce the false positive rate and then induce the toxicity will be reduced a lot. Second thing would be we identify subtypes, the cancer cell subtypes that you want to remove that most aggressive and then most progressive the cell types, right? But actually in the bulk tissue, probably it's not easy to separate that those early stage, late stage tumor cells. But out of the separation by the single cell or spatial data, maybe you can focus on that some late stage specific targets. So you can also select that more important or more the serious targets for the cancer patient. Sorry. Okay. And then also we validate using the protein analysis combined with the RNA and protein together in the same slide. So maybe that is important for checking that sometimes that is the some cleaved or some modified and degraded rapidly, and then RNA level cannot represent that level of protein. So we double check and then whether they are localized in the membrane or cytosol. So if I summarize, actually, we're starting from the old targets, potential targets. That means 20,000 old genes, and then screen off targets with the toxicity, and then prioritize based on our own the method, and then the spatial the features we also combine, and then finally we validate with the protein level. So if I summarize, our context would be first one be a tumor normal separation. It's important for the toxicity, remove the toxicity. And second be a subtype mapping, and then to identify all coverage or specific stage specific the targets. Also, we include the spatial features like as I showed in the 6-line positive cells, that some important feature for the localization, the space, and also multi-layer, the protein level and the RNA level demonstration. So based on multiple data, we found these types of founding model because it's not, maybe you can analyze the RNA-seq data and single state data and the spatial data and the clinical data together, but actually if you combine all the data together, and then more powerful to also mobilize all the data together. And then our direction will be there's a 2 direction, one for the basic concept be we want to know the resistance mechanism and also cancer evolution. So based on this concept, our AI model to direct to consider that kind of concept, because we have the pre-post match samples and then treatment the responder non-responder samples, so we can direct that kind of direction to make a model. And also spatial ecosystem means we can consider not just for the expression, also for the location. So we can consider, another parameter to make a new model and the new targets based on the spatial features. So final slide, actually we are now collaborating with the 6 hospitals in Korea and Japan and also in the United States. Also we're collecting more data, and also especially we are focusing on the treatment response data. And then also we are powering the AI, and then also happy to invite you, any hospital and any institute, also any scientist to develop the model together. So if I summarize that spatial omics data is now, as you know, NGS will be starting from the research and the technical development, but actually now in the routine test, also you can use that information for drug development, other purpose. So spatial omics will be now underdeveloped, but actually will be utilized for the clinical purpose and also other industry purpose. So I think maybe you need to prepare that what you can do with the next stage of the spatial omics technique development. And we joined the Scrum Japan, there'll be large size code. Happy to use that information in the as a consortium. And then I showed that some evidence we did, how we can use that potential new targets for the TNBC cancer. And also, we established that we name it IntelliMed, but actually we are developing this pipeline, I mean the platform, to use that spatialomics data to propose that new targets based on our own model analysis. Okay, I want to stop here to thank our members. Thank you.
Thank you, Dr. Park. If you have any questions, please raise your hand. 会場からのご質問は日本語でも結構です。ご質問いかがでしょうか。どうぞ。今マイクをお持ちします。
Thank you very much for your talk. I just want to understand a little more scientific, situation about TNBC, the spatial, expressions of different, genes and different types of cells. So as you may know, the break of the breast cancer from invasive to non-invasive to invasive. Maybe there is some, the battle between the tumor cells and the immune cells. So in terms of the spatial, analysis, the timing is very important. So is it the pre-cancer or non-invasive status or invasive status, or right before treatment or right after treatment? So do you have any insights around this area about maybe not the product development, or more scientific, the tumor, kind of evolution question?
Yes, actually, the beauty of the spatial omics will be we can identify that area in the same patient, some dysplastic area, and then the early stage and the late stage. So together, and also we can see some interaction between the tumor and the microenvironment. So we can separate that, right? So and then we analyze that evolution of the tumor and also microenvironment together. So, but not for the individual, every individual cases, because we should have the old information in the one slide, right? Yeah, but sometimes you can see
from the population, base.
Yeah,
you can do the cross-sectional,
right. So
comparison and
yeah,
analysis.
if we combine all the data, and then for the example, we have the 200 cases for the treated breast cancer, and we can identify the in situ carcinoma in situ, and also the aggressive and invasive cancer cells, and then we map it in the slide again, then to see any difference between the microenvironment around that area, right? So we can see that is the important, the things for the spatial omics data analysis.
So did you find something new, or?
Yes, we are. We now in the submit the paper in the for the esophageal cancer. So it's the dysplastic, and also so we do find that really dysplastic area they show the high immune response, right? So that, but actually our hypothesis that the immune response they really turn into the adaptive immune, then we can suppress the cancer. But actually, most of the dysplastic area be showing the innate immune inflammation, right?
Thank you, Dr. Park.
Okay, thank you.
Then we have the last presentation by Dr. Alan Nelson, MD, MBA, FACP, Health Sciences Assistant Clinical Professor, Division of Hospital Medicine, and advisor to the Institute for the Global Entrepreneur, IZE, UC San Diego. Dr. Mozart, please.
Thank you. Thank you, everyone. Thank you for having me. This is my first time in Japan, and I'm very excited. My talk is going to be very different from the three previous talks. We're going to talk about some systems processes in the United States, and I'm going to do my best to explain to you the complexity of, it's a very different type of complexity than the complexity we've been talked about in the 3 previous talks. And I hope you don't find it too difficult. So without further ado, so I'm just going to give you a very basic view, a vision of what I think medical practice is going to be in the next 10 years. Unlike the previous speakers, I am a practicing physician and I see my patients in the hospital. And the face of how I practice, I think, is going to change. And this is very pertinent for people who are developing new therapies and tools that are going to be used in the clinical space. We've been talking a lot about omics, different types of spatial omics. We've talked about ways to use machine learning to differentiate renal cell carcinoma responders and non-responders, and then how to gut microbiota will affect mutations in people developing gastric cancers and colon cancers. Where I practice, multi-omics will be used a little bit in different contexts. We are looking at individual patient mutation, germline mutations, and that data is going to be used to make predictions on a patient-by-patient basis, not necessarily for drug development, but for how to stratify risk of our patients to develop various different types of diseases, not just in the cancer space, but cardiovascular, and also dementia is a big area. A lot of people looking at Alzheimer's disease as we become an older planet across a lot of different industries, different countries. So omics is one pillar of this new way of practicing medicine. And the second that we're working on that I'm very excited about is continuous monitoring. These are technologies that are being developed, and you're seeing the very first iterations of this in consumer products that are available, like Oura rings and Garmin watches. But I think this is just the tip of the iceberg. These continuous monitoring technologies are going to advance to the point where we're going to be able to monitor biomarkers on a continuous basis. So I'll give you an example. At UCSD, where we work, we're already looking at a continuous lactate monitor. For those of you who are familiar with the treatment of sepsis, knowing lactate on a minute-by-minute basis is a very important thing. But that's just also the tip of the iceberg. There's also other things, continuous CRP, continuous interleukin monitors, and these are all things that are coming down the pipeline. So you have continuous monitoring, you have omics, and these are 2 very high data fields and technologies. The thing that is most important that I want to talk about is the thing that people need to keep in mind is you need to be able to integrate that into a clinical workflow. So you have a doctor who's practicing medicine, who's seeing patients, and they need to be able to parse through this data and make it usable and actionable for their patient populations. And if you're developing a product, whether it's a therapeutic, whether it's a device, part of the design of that solution needs to be how is it going to integrate into the clinical workflows. Now, I'm going to explain how the American medical system works. It's a very unique system that's different from a lot of the different systems out there in the world. And the other thing I should mention before we go on is because these are high data-driven spaces, continuous monitoring and omics, you have to create an AI solution for a doctor to be able to use this technology to treat their patients. There's a book that's out, Intelligent Health. I'm not related to the author or anything. I think this explains this concept pretty well if you have time to read it. By the way, I have no financial disclosures, I forgot to mention. So we have been talking about a lot of the technologies, spatial omics and using machine learning to differentiate responders and non-responders for cancer. And hopefully that will lead to the development of drug therapeutics. That's ultimately what the presenters are thinking about. Now, whether you make a therapeutic or whether you make a device, the bottom line is if you want to enter the United States, which is the big market in the world, you need to have a strategy for entering that clinical workflow. So you need to know how patients are treated and how the system works in the United States, not just from a medical standpoint, but from a reimbursement standpoint and a clinical workflow standpoint. So in the ideal world, your solution will be seamlessly integrated into the way a doctor takes care of patients in their workspace. I advise a lot of Japanese companies that come over, and you guys have very great technology, very cutting edge technology. The thing that always trips up these teams that come to learn about the medical space in the United States is their ability to integrate that into the workflow of a doctor. The sophistication of a biosensor will only go so far if no one's able to wear it, and that is predicated on how you make a strategy for having doctors use your technology. So the two things that I want to talk about in this presentation are clinical integration strategy and then a reimbursement strategy. You need to know these things, whether you're making a therapeutic or a device, to be a successful entrepreneur in the biomedical space. And I will hopefully, So this is our United States medical system. So I don't have the complexity of the three previous presenters, but I have my own complexity. This is a very complex system, and it takes, trust me, my medical students and residents that I train at the hospital, it takes them years to understand this. And usually I have 1 hour to explain this system and how it works to my Japanese colleagues that come over. I only have 20 minutes with you guys. So I'm not going to go into very in-depth of how this works. I want to give you some broad stroke understanding of this so that you have at least some understanding of where to start with this process of the best way to the irony of this is the best way to understand that complex is to understand this one graph. This graph explains our medical expenditures in the United States relative to our GDP. And in this year, we're now at 20%. So 20 cents of every dollar that's spent in the United States industry is in healthcare. And you might think, well, why is that a problem? The more we spend on health, the better. The problem is this. This is a chart, we call it a chart of the century. If you go on Google and put chart of the century, this comes up. This is a chart that was made by the American Enterprise Institute. And for those of you here that could see my laser pointer, so this is how much we're spending on hospital services and healthcare in the United States. And the important thing is our average wages for an average American is not keeping pace with this expenditure. Why is this important? Well, let me talk about this. So the top bar is how much we spend in the United States. And then this is an average of the other industrial countries in the world, Japan being part of it. And you could see across the board, whether it's our providers, how much cost they are, whether it's our administrative costs, whether it's our drug prices, or whether it's our everything else. The only thing we don't spend more on, which is ironic and sad, is preventative care. So we spend less on preventative care and we spend more on everything else. It's a sad state of affairs. This is a breakdown of so to the credit of you and your country, you're one of the best, most cost-effective healthcare industries in the world. And this blue bar is basically the same bar that I showed on the previous slide. The reason I'm using this is to show that about 60% of the increased spending we have in the United States is because of increased cost of everything across the board, the cost of hospitals, the cost of doctors, nurses, and drugs. And then the other 40% is made up of higher utilization. So we use more healthcare, we get worse outcomes, and these are all important because I'll get to the point of why I'm presenting all this data. This is my most important slide in my presentation. If you could remember this slide, it would help you a lot. This is the hierarchy of what is most effective to enter the United States. And a lot of people that come to our program from LINK-J, they're in this category over here. They have a product that is great, that works well, that increases the quality of care, but it costs more. And believe it or not, you will be less successful than someone who's at the top here, who's increasing quality, maybe not as much, but you're reducing costs. Reduction of costs is on the lips of every healthcare administrator and hospital CEO in the United States, especially right now with what's happening in Washington, D.C. So if you can show that you are reducing costs, you will go much further as a commercialization entity than if you are developing a new therapeutic or a device that increases outcomes or improves outcomes. There's also another book, if you have time, it's a very thick book that explains this concept called value-based care. And value-based care is basically how to increase your ability to provide quality care while reducing costs at the same time. And it talks about how to structure a payment system in the United States to incentivize that type of system. What's happening in the United States is our value-based care contracts, and I'll explain this as best I can in 20 minutes, are increasing. So more and more providers, so health insurance companies in the United States, including Medicare, which is our government sponsored, and Medicaid, are developing what's called value-based systems of how to pay doctors to improve care while reducing costs to the United States healthcare industry. I don't have time to explain how that is. I wish I had more time. If you come to San Diego, I could give you the full curriculum of why this is happening. But to suffice it to say, there's 4 ways that money goes from Americans to healthcare providers. One is through direct payments, so out-of-pocket payments. It comes right out of my credit card or my pocket, goes to the healthcare provider. Most people who are workers in the United States get their insurance through their company. And then that is mediated by all these different types of healthcare insurance companies. So if you, for instance, work for McDonald's or you work for Target, they have a contract with health insurance A, B, or C, X, and then that's how those health insurance policies cover our workers. Then there's 2 categories of patients that are outside of this. One are our Medicare, which is this column, this vertical here. That's through federal taxes. And that covers our 65-year-olds and above. And there are some subcategories without getting too complicated, but just suffice it to say, our 65-year-olds and above are covered by Medicare. And then our individuals who are unable to afford or are not working are covered under Medicaid, which is a different program in each and every state, just to make a complicated story more complicated. So depending on what state you are, your Medicaid will look different. The reimbursements will look different. So the landscape of value-based care will look different based on what state you are in. So whenever I see a patient, 2 things happen. One is I bill for my professional services in the United States, and 2, the medical center bills for their medical center services. And those are mediated by 2 different types of codes. And we try to teach coding to our colleagues that come from overseas, because your go-to-market strategy and your reimbursement strategy is really predicated on your ability to understand coding in the United States. What is a CPT code? What is a ICD code? So the facility, we call it a facility fee. That's one payment that goes from the insurance company to the hospital. And then there's a second payment that goes, a professional fee, to practicing physicians such as myself. The codes that mediate these 2 types of payments are created by completely different entities. ICD codes are inpatient codes. They're based on diagnosis. So you get paid based on a pneumonia rather than what services you provide. And then a CPT code is based on it's called a fee for service. So it's based on what you perform. So if you see a patient, you evaluate a patient, that's a CPT code. If you perform a bedside procedure, that's a different CPT code. Those CPT codes are created by a different entity called the RUC. And then that is a committee that is advised by the American Medical Association. So if you have a new product, I'll tell our individuals that come over, if you have a new product and you want that product to be paid for by this system, you need to have a CPT code for that product. And then that process is a 5-year process, up to 10 years, and that's after you get FDA approval. So you'll get your FDA clearance on your device, and then it takes another 10 years to figure out how to get a reimbursement for that device. So you have to have a strategy for those intervening years of how you're going to pay the bills until you can get reimbursement from our medical system. By the way, this is more pertinent to medical devices than it is to the pharmaceutical industry, because the pharmaceutical industry, you can get payments starting once you have clearance from the FDA. And that's because, again, it's complicated. I don't have time, but the way pharmaceuticals are paid are by 2 different systems. There's an inpatient system and there's an outpatient system. You can start billing for both as soon as you get FDA clearance. The way, let me go to this next slide. And I'll get to that thought in a minute. It's hard to explain it all at once. But one important thing, I added the slide last minute, 5 minutes before the start of the symposium, is another thing to understand is even if you have a CPT code, all you have is a CPT code. That CPT code might not necessarily be linked to a payment. So first you got to get the CPT code, and then you have to convince Medicare to pay a reimbursement for your CPT code. So it's actually an added layer of complexity and bureaucracy to your ability to get reimbursed. Let's see what my next slide is. Okay, so all of this is to say that when you are coming into the United States, you need to have a reimbursement strategy. You need to understand how, not necessarily how are you going to get paid. You need to understand how the physician who is using your product is going to get paid, because it needs to make sense for that physician. So if you're selling a product to a hospital, let's say you're selling a medication, a drug, and this is a specialty drug. A lot of the talks were about oncology. So if you have a chemotherapy drug that you're selling to a hospital in the United States, the hospital will pay for the drug out of their operating costs. That's how it works. But that hospital needs to be reimbursed for paying you for the product. You need to be able to go to that hospital and say, this is the way you get reimbursed for taking care of cancer patients, and this is why it makes economic sense for you to use my product for it to be useful. So there's a strategy, and you might have heard this term before. It's called market access. You need to have market access right at the get-go, because even if you want to become you develop a drug, you get it past phase 2 in FDA, and you now have a strategic partner, let's say Pfizer or AstraZeneca is interested in your product, they won't be interested unless you can explain to them how they're going to get a ROI. And then that means that you do have to understand market access. Well, how big is the market for this medication? How many cancer patients could potentially be treated with this medication for them to invest into purchasing your IP and giving you an exit? In the interest of time, I only have 10 minutes, so I'll skip over this, but suffice it to say that it becomes complicated. How much you get paid is really based on, or how much a hospital gets paid is based on what contract they have with different insurance companies. So you could have the exact same medication, and one insurance will pay X amount of dollars for treating that patient, and then another insurance will pay Y amount of dollars. So it becomes difficult almost to keep track of how much these payments are going to the hospitals, which adds a layer of complexity. And this is why market access teams exist in the United States, because you need these specialists to explain to you how the reimbursement structure works. Come on. There's a delay on my slides. I apologize. This is another important slide. Let me see. Yeah, this is another important slide. There's an assumption overseas that our healthcare industry is a very profitable industry. Maybe the pharmaceuticals are, maybe the medical device sector is, but the hospital system is a margin-tight industry. Our average hospital makes about 4.7% margin based on how much revenue they bring in. So the hospitals, which are the ultimate end user for your products, are running on tight margins. Another reason why I emphasize that reduction of costs is more important than increase in quality at this moment in time, which is why I included those introductory charts. So the other thing in California, in the state that we're from, believe it or not, for every Medicare and Medicaid patient, we incur a financial loss for taking care of those patients. So we get paid 80 cents for every dollar we spend on a Medicare patient, and then 75 on a Medicaid patient. And those make up 70% of our patient population. So on 70% of our patient population in the hospital, we're losing money. So how do we become profitable? It's on that 30%, the private payers, the insurance companies that are from the employers. And if you talk to any hospital administrator in the United States, they're all fighting over that 30%. This is actual numbers. So how much we theoretically bill for, this is actual numbers from UCSD Hospital from 2019. Theoretically, we bill for $8 billion annual in 2019. But how much of it we actually collect is 2.7 billion. Why is that? Because with every contract we have with Humana, with Aetna, with United Health, there's steep discounts. So this is what our sticker price is, so to speak, and the actual money that we collect is this, and our margin is 4.7% of that. Actually, this was 2.9% that year. So suffice it to say, the hospital system, the medical system in the United States is a tight margin. You need to show your end users that there is a cost reduction of your product. These are our expenditures, and you could see numbers 1, 2, 3, 4. These are all people. So our top 4 expenditures are ourselves, and drugs. I can't see from this. Yes. So supplies is where basically you fall. That 619 million is how much we spent at UCSD on medications, all medications, and all of our equipment. So it is actually a minority of what we spend on, and that is commensurate with the other hospitals in the United States. This is an average of how much is spent. So about 60% of healthcare expenditures are on labor, and only about 8% on medications and 13% on supplies. By the way, this is just the inpatient side of things. So this is not the outpatient side, which is a whole other ballgame. The other thing we're doing at UCSD that I'm excited about is getting back to this concept. Omics is one pillar. Continuous monitoring is another pillar. You have to include it into a clinical workflow, and you need AI to parse out that data. Now, I'm using devices as an example because it's easier to understand. So now you want to bring your device into a clinical workflow. What you need is clinical integration. You need workflow integration. You need to know how is that device going to be implanted into the day-to-day work of a doctor and into the ability of a patient to use that device. So continuous monitoring, which is what I'm interested in, will require a lot of data. And you can imagine, let's say I have a clinic and I have 1,000 patients, and on any given day, I'm seeing 25 patients. Well, those 25 patients are all going to come with 6 months of continuous data. And I need a way in a 15-minute clinic visit to give them an action of what to do with that data. So there's a whole brave new world out there of a new type of wearing a continuous glucose monitor, someone's wearing a continuous troponin monitor or any type of monitor. You need a way to give that information to a doctor so that they can use it. And that is a science that has not been created up until now because these wearables have not been a thing up until now. So that's one thing that we're developing, and we're doing this by partnering two of our institutions. We have the School of Engineering, which the dean is here, and then we have the medical center. And UCSD is unique because we have world-class iterations of both of those. So our engineering and our medical center create these workflows so that these products that we develop from academia, from our institution, have baked in to their design an ability to be implemented into a clinical workspace. Let me say that in a different way. We want to create devices that already have the ability to integrate into a live environment healthcare setting from the beginning, rather than developing a device and then trying to bring it in, or we call it shoehorn, and try to shoehorn it into a clinical workflow that is already there. So you could I know you guys were more in drug continuous data can be parsed into usable, actionable interventions. And then that gets put into a health center that is already operating, which we have under one roof at UCSD. And then once these devices or therapeutics are validated, oh, let me say, so we also have machine learning specifically for healthcare delivery. So if we bring to them these continuous data streams from our devices, our colleagues at the center the healthcare center will be able to create, then we will have industry partners bring that to scalability into the market. And then what funds all of this, we hope, is that they will fund our research into developing these products, and it becomes full circle. So they have a great ROI for funding our research instead of going to the federal NIH to try to get research. These devices need to be paid for. And again, this goes back to codes on the books that pay doctors to do continuous monitoring, but it's very rudimentary. There was a question earlier of regulation of AI. Well, it's not just that, it's reimbursement of AI. And this is the Wild West. These are the very early iterations of this. These CPT codes will become more numerous and more nuanced and more complex as the years go by, but we hope that that will serve as the foundation for an industry of continuous monitors. And we need to be able to pay our institution to see patients that are using continuous monitors, and then that is really predicated on the CPT code, and it's also predicated on a strategy of gathering the reimbursements for these CPT codes. So we do have to understand that whole industry. And this is my team of physicians that advises the Institute of Healthcare Engineering, our engineering colleagues. We have a very diverse specialty set here. And with that, I ended right on time.
Thank you, Dr. Moran. We will move on to the panel discussion and Q&A. I would like to ask Dean Pisano to be a moderator. Pisano-san and 3 speakers, please come to, oh, already there, you're there. Pisano-san, please start.
Okay. Hello again, everybody. Let's get the panel session started. My goal in the panel session is for the audience here and online to be asking questions. But to give you a chance to think of your questions, I have prepared a few questions for the panelists. And after I ask each question, I will check, would anyone else like to ask a question? Okay, so the first question I will ask, and I will ask each speaker in turn to make an answer. I'll start with Ludmil and then move down the line. So I will ask this general question. In the work that you are doing, what is the biggest obstacle that you must to bring your findings to actual healthcare and so have impact on patient outcomes? So a short version, what is slowing down a change in patient outcomes from the point of view of your work? What is clogging it? What is stopping it? Ludmil.
That's a great question. And I have a 2-part answer. And the first part is, from a research perspective, is data, the ability to get data, the ability to share data, the ability to analyze the data. But that only solves the research part. That's the biggest bottleneck there. The bigger bottleneck that I have realized is the research is probably 5 to 10% of the work. The engineering is another 5 to 10% of the work. After that, especially in the United States, as it was described so very nicely in the previous presentation, all the other parts that we as a scientists are never trained, the understanding the regulation, the reimbursement, the payment system, they're the bigger problem. So that's, in my mind, the obstacle, the complexity of the subsequent system that comes after the science and after the engineering.
Okay, thank you very, very much. Dr. Nakagawa,
Yes.
you have some
So
thoughts?
yeah, I'm almost researchers, and also I'm also doing the medical practice in a hospital or a small clinic, and I know the both side. So we have a kind of data from basically is of a frozen sample. This is a kind of a standard in the research levels, but I realize it's like the most of the hospital, even in Japan, don't have a reserve stored of such kind of frozen samples. So that's a big hurdle. So I've decided. So now that we are now working on a puffing block, HE block of a sample to get the genomic data or RNA-seq data or special data.
Okay, thank you. Thank you very much. I'll ask Dr. Mawazam to offer his thoughts.
So I don't research like the previous 2 panelists, but I do work with our engineering colleagues to develop our continuous devices. The bottleneck on that is not the innovation from the engineers. The engineers are wonderful at innovating. The bottleneck is the health system needs to create a entry point for these data clinical flows. So the innovation doesn't come from the engineers in my sector. It needs to come from the hospital administrators, and that's what we're working on right now.
Okay, thank you, panel. Oh, good.
Yeah, one more hurdles for us, like standardization of the data. Even in Japan, each hospital has some amount of clinical data. Genomic data. Genomic data is almost standardized, but it's so hard to standardize all such kind of the data, clinical data across the hospitals across the countries.
Thank you. Thank you. Any other thoughts from the panelists? Okay, so my first check. Are there any questions from online? And I thought one or two people here looked like they were ready to ask a question. Are you sure you don't want to ask? Yes? Please, just wait for a microphone so the people online can hear you. Is there someone to deliver a microphone? Thank you for the question from the audience.
So my question is, like, if there is any, emerging technology that, sorry, can you hear me? So if there is emerging technology to replace all labor-intensive workflow using AI, like single AI-based single universal like diagnostics, then who we should partner with in the US to implement in the real workflow? Like if there is any successful case study that industrial player partner with like medical centers or research institute to implement that technology into the practical scene, then I'd like to hear your knowledge.
All right, so the panelists, please, whoever wants to jump in.
Are you talking about the labor of developing drugs and doing the machine learning, or the labor of delivering healthcare?
More on delivering side, not developing side.
So the delivery of healthcare is the domain of the healthcare system. So the question you asked is, who should we partner with? You need to partner with an enlightened healthcare system. So a system where the administrators are open to having outside vendors and developers to come in and develop tools for their health system. One of the biggest impediments I see, especially for our Japanese colleagues, is they create these AI tools, they create solutions, apps, devices, and then they come into the United States and they try to give it to someone. But those clinical workflows are so sclerotic. They're so difficult to enter that it makes more sense to find a partner that is willing to let you experiment in developing your product. So UCSD is one of those places, but there aren't too many of them in the United States.
Thank you. Is there a question? Yes, a question from the audience. Right up here in the front. Thank you for waiting for the microphone.
Sorry, I'm asking a lot of questions. So it's a similar question to you, Dr. Moazam. Just give me explanation. One example, if you do have, is for the, just as Professor Dr. Nakagawa mentioned, the data in the medical record, EMR, is very, very different in Japan and maybe similar in your country. So I just want to understand, I heard from some of the doctors from the United States mentioning that you are using the voice recorder to record the patient, your dialogue with the patients, and automatically generate the record and put into your system. So how was it started? And is any obstacle or whatever problems you have had, and how do you overcome the problems? I think that's one of the way of to increase the proficiency of the delivering the healthcare.
Let me make sure I understand your question before I answer it. So you're asking, how did we implement an ambient scribe into our healthcare system, and then what were the challenges that we had? So UCSD partners with a vendor called Nabla, and that is our ambient scribe solution, and I use it when I see my patients. Again, a lot of it is the credit of Karandeep Singh, who's the chief of our Center for Healthcare Innovation. They have access to our EHR, our electronic health record, but they've opened up APIs to allow innovators to come in and create these solutions for our clinical workflow. And that's exactly the way to do it. I can't tell you how many teams and startups are in the United States, even in San Diego, that have ambient scribe technology, but it's howling into the wind. There's no way to plug it in. You need to plug it into a healthcare system.
So the question is, who was paying for that? I mean,
so UCSD pays for it, and we pay for it because of two reasons. One, it increases our efficiency, and because this one's not working. One, oh yeah, that's much better. It increases our efficiency, and two, it increases our physician wellness. Because you can imagine, an ambient scribe is not going to change our reimbursement. We're going to get reimbursed. No one's going to pay us more because we use the ambient scribe. The value of it is less burnout amongst our physicians. If you burn out a physician, now you have to spend all this time rehiring a new one, and physicians are hard to come by. So that's the value of it.
Okay, any other questions from the audience? All right, I'll ask a leading question, and perhaps that stimulates more questions. Anything online? Okay. So to change focus just for a second, for each of the panelists, Alan, we'll start with you and we'll come this way through Nakagawa. How do you define success in the work that you're doing? And what part of the medical ecosystem needs to hear more about it? So in your work, how do you define I am successful? How do you know you are successful? And who needs to hear about it? A tough question, I know.
That's a very tough question. There's so much to say about that. I want to give the other panelists a chance to speak. The way I would define success is it's no good to have a clinical a device into your workflow if you're not changing end results. So if you're not changing health outcomes. So you need to be very strategic, but also smart about picking the endpoints, the primary endpoints for your solution. So let's say you have a good example, we're doing this right now actually, is a continuous lactate monitor. So for those of you who lactate is something that goes high when a patient is becoming septic. Knowing that on a minute by minute basis is a very valuable thing. It doesn't make any sense to say, oh, we have this lactate monitor. Okay, but what are your sepsis survival rates? If your survival rates are the same, then all you're doing is just using a device for the sake of using a device. You need to show some change in sepsis survival by implementing that. So there needs to be some science that, some health system science that's done when you implement these.
Nakagawa-san.
So, 日本語で、Japanese。MD、PhD、実際の医療に携わってきたものとしては、自分たちの、見つけたことが医療実装されるっていうのが、サクセスフルなもんだとは思ってますけども、ただ、アメリカで、4年間か5年間ぐらいずっと研究してきていて、その辺のところ、なかなかアメリカでは結構やっぱりそういう医療実装っていうのがもうトントントントンと進んでいっちゃうんですけども、なかなか日本では進めないっていうのがすごく今フラストレーションが溜まってきているような状態ですね。それは多分そういう医療実装のシステムなりアプルーバーのシステムがまだまだ日本は進んでないと思うし、あとはコストの問題もやっぱりあるかなとは思います。で、国としてどういったのかな、全体として彼が説明してたように、ヘルスケアシステムとしてサクセスフルなのかどうかっていうのがちょっと少し質問というかあって、先ほどのクオリティとコストっていうような考え方があったりしましたけども、そのエンドポイントとして、クオリティがサクセスフル、高いのがサクセスフルだというよりも、ヘルスケアシステムとしてサクセスフルって っていうのは何なのかなとはちょっと思ってて、どう言ったらいいのかな。例えば日本人の場合は、コストは安く、少なくともアメリカよりもコストはできている。で、さらには、例えば平均寿命っていうことを考えたときなんかは、国としては、やっぱり日本も非常に優れたシステムを持ってるかなと思いますけども、その辺の、どういったらいいかな、国として、そのヘルスケアシステムとしてのエンドポイントっていう、クオリティのエンドポイントっていうのは何なのかなというのはちょっと疑問に思いましたけども。Do you have any comment about the end point of a healthcare system in each patient or for each healthcare system?
That's a question way above my pay grade. Now you're talking a matter of how to compare at the national level the outcome of systems. That is a very people have tried to do this, and this is not my realm. This is the realm of public health, and I think there are markers that you could look at. So QALY, QALY, you could convert health span and lifespan. It's one thing to live until 95 years old, but it's another thing to live vitally until 94 and then have 1 year of bad health. And then, so to convert that into numerical terms, we have QALYs. That's one way of doing it. And then also, I do think how much you spend on healthcare at the expense of education, defense, other things is also another marker that you should take into consideration.
Okay, I'm going to ask Professor Alexandrov to make some comments.
Yeah, I think the question was, how do we define success? And I think, well, I'm a basic science researcher. So from my perspective, success is we'll have some discoveries. Some of those discoveries will inform us about the world. Others will be useful for patients. For those that are useful, my job as a researcher is to prove that it's useful enough, to de-risk it enough so others would want to take it, commercialize it, and put it within a system. So my job as a researcher is to get to the point and say, hey, I have something interesting here that can help patients and something that others would want to raise risk capital to actually commercialize and put forward. So defining success for me is actually getting to that point, getting fundraising over an idea and then leaving it into the hands of people who know better than me the healthcare system to actually commercialize it and put it into use.
Hey, thank you. Thank you. Okay, I'll give the audience a chance. Is someone thinking about, okay, we have one. Please wait for the microphone.
Thank you very much for your discussion. What's the future perspective of the precision medicine from each panelist? Maybe the 2030? Thank you.
What does 2030 look like? There is no wrong answer, remember, because no one really knows. So make a wild guess, each one of you.
I would say anyone who's trying to make predictions more than 10 years, just throw that prediction in the trash, because beyond 10 years, you shouldn't be making predictions. But 2030 is a good time point. My slides I presented, just to answer a way to answer your question, me personally, I looked at my 23andMe, and I looked at all of the things that I'm at risk for, and these are all direct to consumer because they're not in clinical workflow. So 23andMe, I looked at something called NeuroAge, which looks at my risk of developing dementia, a few other things. So that is my omics pillar. I also wear an Oura ring and I wear my Garmin and I try to hit my marks. So that's the continuous monitoring. What I did was I tried to put all that data in. I put it into Claude, believe it or not. I have Claude as well. I love Claude. I put it all into Claude and I said, make me a comprehensive plan for my health. And it nailed it. It knocked it out. So I think that is what it's going to look like. We need to have our genetics fully investigated to give us actionable data, continuous monitoring on top of it. All of that has to be parsed through effective AI, and then that has to be integrated with a clinical workflow, a doctor to give you answers. So that's the answer to 2030.
Okay, other panelists?
Yeah, so it's probably dependent on the countries. I don't know what's happened in like United States or European country. It's a typical different situations. But when I focus on Japan, I'm sorry, it's not in the progress so much, probably 4 years and 5 years, because we can do get the data of the mixed data of genomic data of each patient, and but it's like probably it's kind of a cost. When I think about the medical implications, we have to think about the cost. So in Japan, we have I'm proud of uniform public healthcare system in Japan. So it's very working well, I hope, and in the future. But the healthcare system in Japan, it can be the set up the limitations of the healthcare cost. So even panel sequencing for the cancer, just like 5 or 6 years ago, it's just like approval. But still there, the cancer, panel sequencing of cancer tissues of the CGP panel, just like a cover of the patient, of a cancer patient after the first line, second line treatment.
Okay, Professor Alexandrov.
Yeah, a very thought-provoking question, because as it was said, it's 4 years away. It's close enough for us to be able to make predictions. And what I can come up with is, at the moment, there is the promise that AI will change everything fast, and will revolutionize almost every single aspect of precision oncology, of precision medicine. To be frank, I think that promise will fail by 2030. And I think not much will change. And I say that with sadness. I think we'll see exactly the same thing that we're seeing. Maybe it's a few more drugs, maybe a few more biomarkers, but no fundamental change.
Okay, any other questions from the audience? Is there another question? Yes. Here comes the mic.
Sorry, question to Professor Alexandrov. Maybe going back to your presentation about the deep HRD, and you just mentioned nothing changed too much in 5 years or 4 years. I just want to understand, is it any possibility that the AI teach pathologists in the future or in the very short term? Because the pathologist is seeing the from the microscope, classically identify the cells and maybe the grade of the tumor or whatever. So, and that's the way of they're doing. So AI is being trained or machine learning and learn something very differently, and maybe something of not explainable, but it linked to the nature of the the cell or the tumor. So is it any possibility that from the AI perspective, you teach the pathologist that you should see the specimen this way, and you can find something new, and that can rewrite the textbook in the future, if possible.
I think it's possible to find new information that's important, that's relevant. I think pathologists are very resilient creatures. I think teaching them something new that they haven't been taught is hard. I think digital pathology has been around for now maybe a decade or more, and integrating it, even in the top centers, has been very, very hard. On one hand, pathologists fearing AI for taking their jobs. On the other hand, them not feeling qualified. So I think there is huge value. I think the process will be substantially slower. I think, especially in systems which are that conservative as the healthcare systems, I think it will be just much more glacially, painfully, unfortunately, painfully slow process.
Okay, is there another question from the audience? All right, so I'll ask a different question coming from a different angle. So first off, to be a dean of a school of engineering, you have to be an optimist. Otherwise, you quit the job. Okay, so all right, the pessimists commit suicide or leave. All right, the optimist. So I will now ask an optimist question, which is, I believe that the healthcare systems will receive many pressures to change. Either it's a financial pressure, or it's a demographic pressure, or it's some other pressure. There will be pressure to change. So the question is, and again, we'll let any panelist volunteer to go first or second or whatever. Do you see changes coming, one, from outside disruptive agents such as entrepreneurs and startups and new companies, or two, from deliberate processes within? So what do you think will be the way healthcare changes? Will outside agents change it, or will healthcare decide to change itself? Any thoughts?
Hello. Yes.
Yep.
That's an awesome question. I think that the innovations that have been had up until now were from the entrepreneurs and the small fries. But now we're getting to a point where those low-hanging fruit have been picked, and now we need to make fundamental changes in our health system so that more entrepreneurs of the future can. You can't keep relying on entrepreneurs to innovate through problems in the healthcare system when the healthcare system itself needs innovation. So an example of this, if you want all these continuous wearables to be brought to bear, you need to create a health system that lets you plug them in. And that doesn't come from the engineers, that comes from the health administrators and the policymakers. And the policymakers need to also create reimbursement strategies so that these become financially viable solutions.
Okay, other thoughts from within or from outside?
Okay, this is kind of a very, very political comment. So I just like to talk to Japanese. Okay. 先ほど言いましたように、日本の健康保険システムっていうのがやっぱりどうしてもそういうイノベーティブなものに対するまあチェンジですねっていうのをなかなか引き起こすのは難しい。少なくともアメリカに対して引き起こすのは難しいっていうのはやっぱあるとは思うんですよね。で、なので、それであれば、まあ、なんか技術的に問題ないと、アプルーバルされると、PDMA だとかアプルーバルされれば、もうあとは、自費診療の方に持っていくっていうのが多分一番、ストレートというか、一番そのチェンジを引き起こす可能性があるんじゃないかなと思います。日本のそれは多分、日本のヘルスケアシステムの中で、以前に、ブラカのパネル検査ですね。ブラカ1、2の検査の時でも、その辺のところはね、ちょっと議論したことがあって、まあ、ブラカの、シクエンスに関しても、ようやく去年にアプルーバル日本でもアプルーバルになったんですけども、その前はなかなかならなくて、もうそれだったらやっぱり自費でやったらどうだって、または、日本はプライベートのヘルスケアシステムもあるので、そのプライベートのヘルスケアじゃなくて、保険制度ですね。個人保険の生命保険だとか、そういうふうながん保険だとかあるので、そういうのでカバーしてもらったらどうだっていうふうな意見もありました。
Okay, thank you very much. Professor Alexandrov, you want to toss in a comment?
Sure. So first, I realize I'll never be a dean because I cannot be as positive. Being from Eastern Europe, especially, there's always gloom in everything we see. I agree with what was said. I think the innovation will come mostly from the inside. I think from the inside, there will be the pressure of inefficiency in this, the large healthcare systems, whether they're national levels or whether they're universities, the Byzantine type of inefficiency will start bringing them down. And the new technologies coming from entrepreneurs will be the only way for some of those systems to survive. And the ones that adopt it will survive and the other ones will disappear.
Okay, so it's time to close the panel. I want to thank all the panelists for their wonderful answers. We tossed some tough questions at you, and I think you did great. Audience, would you like to give our panelists a round of applause?
Thank you for all panelists. Finally, we will move on to the closing remarks. Panelists, please return to your seat. Firstly, we will have Dr. Ikuo Hayashi, President and COO, LINK-J. Okay, Hayashi-san, please.
はい。では、日本語で、クロージングリマークさせていただきます。本日はですね、第8回となります、LINK-J、UCSD ライフサイエンスシンポジウム、ゲノム解析、AI、個別化医療、次世代バイオマーカーと医療応用に、ご参加いただきまして、誠にありがとうございます。主催者を代表いたしまして、LINK-Jの林から一言ご挨拶申し上げます。本日はまずご登壇した先生方、そして UCSD の皆さん、そしてご参加いただいた皆様にまずは御礼申し上げます。また個人的にもですね、UCSD のアラムナイの一人としてこういったイベントに携われることを非常に嬉しく思っております。で、本日は4名の先生方ですね、アレクサンドロフ先生、中川先生、パク先生、それとモアザム先生からですね、ゲノム解析と AI によって、まず、疾患リスク予測、早期診断、治療選択、創薬、そして、医療実装まで、どうつながっていくかということについて、大変示唆のあるお話をいただきました。また、パネルではですね、非常に、プサン先生の方からタフなクエスチョンというような方がありましたけれども、まず、研究から、ソーシャルインパクトを生むまでに、どこに、ボトルネック、オブスタクルがあるかというお話からですね、それから、そういった活動を通じた、成功、サクセスというのが、一体、どういうものなのかということで、まず研究の立場から言えば、ディスカバリーであり、医療の立場から言うと、ペーシェントのアウトカム、そして全体を見れば、社会全体のヘルスケアシステムが改善されるという、そういったお話もございました。また最後にはですね、社会を変革するようなイノベーションがどこから来るのかということで、ここはインサイド、アウトサイドどこからかというところでいろいろございますが、そういった中で、我々LINK-Jのようなエコシステムコミュニティがどういった貢献ができるかということを考えさせられる、リアル会場ではネットワークの方がございます。今回の、本日の議論がですね、今後の皆様の研究事業、あるいは新しい連携のきっかけとなりましたら、主催者として大変嬉しく思います。本日はご参加いただきまして、誠にありがとうございました。
Thank you, Hayashi-san. Then we will also have Ms. Miwako Wanga, Senior Director, International Outreach Office of Research Affairs, UC サンディエゴ。Wanga-san, please.
林さんがもうすごい素晴らしいまとめをしてくださったので、何を喋ろうかと思いますけれども、LINK-J さんとはここ10年いろんな仕事をさせていただいてまして、このシリーズも8回目となりました。今回は本当にカッティングエッジなすごく上流の研究からそれを実装するためのテクニック、特にアメリカにおける実装をどういうふうにするかっていうところまで割ともう上流から実装までっていう形でやりまして、皆さんお楽しみいただけたのではないかと思っております。UC サンディエゴの方では日本の大学とかアカデミアと非常に緊密にコラボレーションしていまして、こういう日本発のイノベーションをアメリカで開花させるためにはどうしたらいいか、トレーニングプログラムをいろいろ提供しています。ぜひもうこの素晴らしいイノベーションを世界的に 実装するぞという意気込みのある方は、ぜひサンディエゴにいらして、私たちのプロフェッショナルと一緒にそれを実現できるように、夢を追いかけていきたいと思ってます。最初のその入り口として、このリンク J さんのいろんなイベントがございますので、引き続き私どもも情報発信してまいりますし、ぜひブラッシュアップしてアメリカ市場に来てください。お待ちしております。どうも今日はお疲れ様でした。ありがとうございます。
Thank you, Hayashi-san and Wanga-san.